अभी शुरू किया है, जैसे अभी 5 मिनट में होने वाला है। और तुम लोग क्या कर रहे हैं? अच्छा, ठीक है, तो हम तुमसे बाद में बात करते हैं, ठीक है? क्या होने वाला है ऑनलाइन?
Very good morning.
Hello, good morning.
Good morning, Dr. Mahensin, Dr. Sunil, Dr. Rakesh Bandicho. Sorry for delay. Due to certain unforeseen reasons, we got delayed. But now we are ready. On the second day of the international conference, we, we are going to have a very nice day with a galaxy of eminent speakers. Dr. Mahan Singh, Assistant Professor, Department of Surgery, University of Miami, Miller School of Medicine, USA, has joined us. Very welcome to you, Dr. Mahan.
Thank you.
Dr. Mahan is a seasoned molecular and cell biologist with more than 20 years of experience in cancer biology, signal transduction, genetics, and molecular epidemiology. He has experience with CMC development, including validation of GMP and non-GMP biases to support extensive experience in identification and characterization of new signaling proteins involved in cell cycle, apoptosis, cell migration, invasion, and cell therapy using cell culture and animal models of the disease. He has experience of supervising preclinical research projects aimed at studying underlying mechanism of new investigational cancer therapies in various cell-based animal models of gastrointestinal and breast cancer. He is a specialist in cancer research, drug development, preclinical studies, signal transduction, cell biology, stem cells, and human genetics. Dr. Mahan has a long-standing research in this field, very good researcher and very good speaker. Welcome, Dr. Mahan, in this morning session as a first speaker of this conference of second day in the morning. Welcome, Doctor.
Thank
I
you very
invite
much.
you. Yeah, I invite you to present your keynote address on this.
Thank you, convener, and everyone in the audience. So today I decided to talk about something that is very relevant to India. Although my field is in, like Dr. Pandey mentioned, I have been researching in the field of cancer biology for a long time. But this particular cancer called gallbladder cancer is very relevant for India because it is found only in few countries, and India is one of the country where it is found in large numbers. And I chose this topic, although I work on several other aspects of the cancer, particularly genetics and cell biology. But this is a cancer is very cancer which is very dear to me because I started my PhD back in India in Lucknow working on this cancer. It's been 17 years I did my PhD. I came to US. I have worked on different aspect of cancer biology, but this thing never went away from my mind because this is something which has still been ignored largely by world scientists only because this is not common in Western world. And in India, I heard that since I left India, some of the researchers have started working on it, but it is still not there because of the lack of awareness about this thing. So please, I would suggest you to go ahead and stop me because this is a topic that some of you may not be very well familiar with, especially the technical terms and the jargons. Please stop me whenever you want, or anything which is going beyond scope, and I can explain it in a little simple thing. Like I said, I have been a cancer biologist for a long time, and this one is something I just picked up on one aspect of drug development. regarding gallbladder cancer. So I'll go through some of the slides and then you will understand what is the problem. So gallbladder cancer is one of the most common biliary tract cancer, yet it is rare. Rare in America, in Europe, and most of the developed countries. Not in India, not in China, and some of the Latin American countries where it is found very much. India, in particular, is a high incidence area that contributes
Slide
10%.
share का option दे दीजिए.
Please go ahead. Oh, I did not share. Sorry. Sorry about that. I did not share it. Uh, can you? So, can you see the screen? Can you see the screen? Okay. So, hold on. Yes. So, is it visible to everyone?
Yeah.
Okay. So, like I said, gallbladder cancer is one of the most common biliary tract cancer, yet it is very rare in Western world, particularly in America and in Europe, where most of the cancer research, state-of-art cancer research occurs. India in particular is a high incidence area that contributes to total burden of global gallbladder cancer incidence up to 10%. Why it is important?
Okay.
Because median survival time for patients with gallbladder cancer is limited to 1 year. Now, most of you would know that a lot of cancers, for example, breast cancer, colon cancer, these are the cancers when they are diagnosed. You can, if you are diagnosed in time, most of the time you are going to, survive for, depending upon what stage the cancer has been detected, you are going to survive like somewhere between 5 years to 10 years, 15 years even. But gallbladder cancer is one of the rare cancer which people don't survive more than 1 year once it is diagnosed. Surgery is the only curative treatment. Unfortunately, it cannot be offered to most of the patients due to late presentation. Means when people come, it is already late for surgery, and most of the chemotherapy don't work on it. So palliative chemotherapy means chemotherapy that is offered only to treat their symptoms or reduce their pain and all that is limited to capecitabine and 5-fluorouracil oxaliplatin and gemcitabine based regimens, which actually don't work. Means they just reduce the pain, but they don't really help patient treat the cancer. So despite some advances in understanding of molecular determinants of the GBC pathogenesis, hold on. So I'm having a little trouble here. Yes. So despite some advances in understanding of molecular determinants of GBC pathogenesis, development of the targeted therapies for GBC is still far away. So as some of you may know, these days, people, when they are diagnosed with cancer, they are not only offered uh, drug therapies, they are also offered with targeted therapies. Means therapies which are targeted for their special molecules that are specifically abrogated in that particular kind of cancer. And these are the therapies which have been very helpful in the treatment of cancer in last decade or at least 2 decades. But unfortunately, that cannot be offered for the gallbladder cancer patients because targets for the gallbladder cancers are not known. Okay. So geographic distribution, if you look at it, you see one of the country which where it is found very highly is Chile. And in Chile also a particular type of tribe that is called Chilean Mapuche Indian females. That is where it is found the most. You can see there are a couple other types of the people or countries or ethnicity where this one is found more common. Let's come to India, where in India also it is not found everywhere. If you talk to anybody, especially a doctor that comes from South India, they would tell you that they haven't really seen any gallbladder cancer patient in their clinic. because it's not found over there. So what has been found that most of the time gallbladder cancer is concentrated in an area which is actually called Gangetic belt, means places which are near to the Ganga. So starting from Punjab, so Punjab, Delhi, Lucknow, where I worked as PhD student long time back, Bihar, Varanasi, West Bengal. Besides, it is Maharashtra where it is also found, but it is mostly because the people from Gangetic belt they migrate to these areas, and that is why what they have noticed that the people who are living in Maharashtra but originally migrated from Gangetic belt are the one who are more susceptible. And there are, for some reason or other, which is not explained very well, in Chennai also they found some patients. But like I said, most of the patients are found in Gangetic Belt in northern area, northern India.
Yes.
Okay, so risk factor for gallbladder cancer. There are many. Some of them are well defined. Some of them are not. Two risk factors are most important. Just don't go through this whole list because these are some of them are very complicated, and it will take a lot of time to understand. But the most obvious risk factor for gallbladder cancer is gallstones. So, like you may have heard that people talk about, oh, somebody has gallbladder stone. So gallbladder cancer, and most of the doctors, even if you talk to them in India, they will say that, okay, gallstones, long-term persistence of gallstones is actually leads to gallbladder cancer. But that's not true. As a researcher, I know, and the people who have seriously been working in this field, they know that gallbladder cancer, if gallbladder stone is one of the causative factor, associated factor for gallbladder cancer, but it does not necessarily cause the cancer, because in Europe and in America, the people they have more gallbladder stones than in India. But here, people develop very less number of people develop gallbladder cancer. So it means that gallbladder stone, the presence of stone in the gallbladder, may be one of the associated factor, but may not be the major reason. So what is the major reason? Major reason, if you look at it, there are some of the bacterial infection, especially Helicobacter pylori and Salmonella carriers. So coming back to Salmonella, some of you may already know that Salmonella is one of the bacteria that causes typhoid fever, and typhoid fevers are very common in India and in some of the Latin American countries also, like Chile, where gallbladder cancer is very common. So it was my PhD thesis long time back, and I was working on it, and we had found that Salmonella carriers means the people who ever get typhoid fever, 1% of them become chronic carrier for that. It means that 1% out of 100 people who will ever get typhoid fever, they will harbor asymptomatically this bacteria in their gallbladder. So asymptomatically means these days with COVID 19 situation, most of us already know what asymptomatic means. It means that the people who don't really show any symptom, but they have the bacteria. In case of COVID, they have the COVID virus. So asymptomatic carrier of Salmonella means people they harbor bacteria, but they don't really have typhoid fever. So 1% people who become carrier for Salmonella, actually Salmonella goes and resides into their gallbladder. That's one reason that it doesn't cause the fever, but it stays right there and proliferates, keeps proliferating. So the idea is, and some of the people, and including me, have proven before that, but it's not completely proven, have shown given some proof that Salmonella bacteria may release some kind of factors, some toxins that, along with the presence of gallbladder stone, may inflammate the inner lining of the gallbladder, and that actually leads to inflammation and metaplasia and carcinogenesis. That is the concept that some of the people in the world are working on, and there is some proof to that, not completely proven though. So this is the risk factor. Anyway, some of my gallbladder cancer work, like I said, I used to work on this cancer long time back, during my PhD days. So back in those days, I was the first person from India who published the spectrum, mutational spectrum of KRAS oncogenes. So some of you already must know KRAS is an oncogene which is very important for a lot of cancers. In gallbladder cancer, it is also important. And from India, my report was the first report in 2004, where we published that it was actually mutated in 35% patients. Then we also showed one of the gene polymorphism called apolipoprotein that was very common in Indian population, especially the patients near Lucknow, because that were the patients who used to report to our center in SGPGI, Sanjay Gandhi PGI. Go ahead. Did you say something? Okay. So what we shown that one of the gene polymorphism called apolipoprotein that makes people susceptible for gallbladder cancer. How exactly it does that? Nobody knows. Not even after 16, 17 years after my work, nobody had shown it. But we found that this particular gene polymorphism can put the people in risk for development of the gallbladder cancer among those who already have stones. But recently, in last couple of years, even though I am living in the US and we have a lot of difficulties in working on this cancer, I started working back in the last 3-4 years on this cancer again, and I'm collaborating with some of the people back in India and in Chile also. Okay, Yashwant Pandey, go ahead and ask your question, please. Do you want to ask some questions? Yashwant Pandey, please allow Yashwant Pandey. Hello. I think he wants to.
Any question may be taken after the talk.
Okay, sure. We can talk about that. Sure. So. Okay. So what are the challenges? There are a lot of areas in gallbladder cancer. Some of my collaborators and I know people in the field are working on, but my specialization is in the drug development. So I thought what we could do to facilitate the research in this particular area, especially given the fact that we don't really have good drugs for gallbladder cancer. Only thing we can offer or the doctors offer to the patient is chemotherapy, and chemotherapy doesn't work. It only helps them alleviate the pain. That's about it. So why is the problem that what is the major problem that people could not develop drugs for gallbladder cancer? Because not many druggable targets have been identified. So like I said, for many other cancers which used to be almost untreatable 20 years back can be treated today because of the development in the area of cell and molecular biology. People have been able to identify the targets in that particular cancer type, and now, since they identified the targets, they have been able to develop the drugs or repurpose the drugs which were already available in the market to use them in that particular cancer type. In gallbladder cancer, we don't know about that. In recently published two big genomic and transcriptomic studies, one from Japan and another one from China, in last, I would say 3 or 4 years, they were only big studies where they have shown that ERB2 family members, specifically ERB3 and ERB2, have been shown as major players in 35% of the cohort of the GBC patients. So this is one of the target. I don't know if any clinical trials are going on or not, because in US there are no clinical trials going on because we don't have enough patients. Major patients are in India, some in China. I'm not aware of any Chinese studies, and as far as I know about Indian people also, they are not probably doing any clinical trials. Although we have the drugs, very good drugs available for ERB2 family members, especially ERB2. So many drugs are available, which are used to treat other cancers, particularly breast cancer. So what can be done, and people who are listening here and have interest in this field, they might think about it, that why not try these RB targeting drugs to treat the gallbladder cancer patients. At least they could, especially scientists, I know that there are no doctors in this meeting, but the scientists, they can think of testing those things in cell lines and other model systems and see how they have effectiveness in gallbladder cancer. Anyway, So another example, important mechanism for gallbladder carcinogenesis is P53 mutation and c-Myc amplification, at least in a subset of chronic infection of Salmonella typhi. So there was a paper from actually Varanasi some time back. It was not exactly from Varanasi, but one of the person, a pathologist who works on gallbladder cancer, he collaborated with people in Europe, and they published, it's a very good study where they first time shown that the people who have Salmonella type infection, like I said before, they also have c-Myc amplification. And people who work in the field of cancer biology, they probably know c-Myc is, another important oncogene, just like KRAS, which is associated with most of the cancer types. So that was a good thing because there are a lot of people in the field who are trying to target c-Myc, although it is not that easy to target c-Myc, but people in especially in pharmacology field, people have been like trying so hard for, they have invested their decades of their life in developing new therapies to target c-Myc related amplification in various kind of tumors. Anyway, so I was talking about this cancer, and like you could see, typhoid fever is very common in this particular part of the world. India is one of them, you see. And fortunately, I mean, coincidentally, these are the places where gallbladder cancer is also found. That also makes sense that, the people who tend to have typhoid fever, 1% become chronic carrier, and they probably develop in future, not exactly immediately because that takes time, maybe decades, but they are more likely to develop gallbladder cancer. So in that study, they had shown that P53 mutation and c-Myc overexpression was one of the common feature for Salmonella typhi infected tumors. Anyway, and they went on to show how exactly, especially 86% of these patients who were, gallbladder cancer patients and also had typhoid infection, they were amplified for MYC oncogene. Anyway, so in this study, we decided to test if targeting a signaling pathway underlining chronic and Salmonella carrier state could be a therapeutic strategy for the treatment for GBC. So here I'm talking about a compound called triptolide. It is very common compound. People, if you search for it on the Google, you will immediately find about it. This is actually derived from a Chinese herb called thunder god vine, and you can see its structure. And one of the professor who is my collaborator over here, he's incidentally he's also Indian. He developed a water soluble compound of triptolide called minilide. So it's a more, it's a useful for clinical perspective, like you can directly give to the patients if this is ever developed. But original compound triptolide, if you search for it, you will find thousands and thousands of publications, but some of them are in cancer and some of them are but most of them are in other diseases. Like I said, it is one of the herbal medicine from China. So they have been using it for thousands of years, just like, our Indian Ayurvedic system. There are medicines that we have been using for thousands and thousands of years. So they, isolated this active compound called triptolide. And one of this Indian professor, he made another compound so that he could patent it. I mean, because of commercial interest, and also because it is a water soluble compound, so called minilide. So what exactly it is? This is a water soluble analog of triptolide. The minilide thing has been shown highly effective in reducing tumor growth, metastasis, and improving survival in mouse models of pancreatic cancer. So this guy has published a couple of papers, actually many papers in last 10 years, who developed this compound called minilide or triptolide, you could say, because basically both are same. So they have been shown to reduce tumor growth in many types of cancer. Pancreatic cancer, they are advanced to second stage of clinical trial, phase 2 clinical trial. International clinical trial is ongoing. So that is where I started thinking that and since this professor is next, his lab is, it's a big lab and they are next to me. So I thought, what about trying this drug in uh, gallbladder cancer? Because gallbladder cancer and pancreatic cancer, I am a gallbladder cancer researcher, so I know that both of them, they in embryological study, it has been found that both come from the same origin, so they derive from the same kind of stem cells. So I thought, what if, it's likely that this compound is going to be effective in gallbladder cancer as well? So I just gave it a try and see. I would say a little more things. there was another thing with the triptolide, that out of a lot of compounds, one of the compound where it was found in PDAC and some of the other cancers where they have PDAC means pancreatic ductal adenocarcinoma, which is what this, my neighbor's lab, this Indian guy's lab works on majorly, because they are testing this drug's efficacy on pancreatic cancer in phase 2 clinical trial. MYC is also amplified in 20% of the pancreatic cancer. And one of my collaborator, where I was trained in Philadelphia a long time back, actually Dr. Rakeshwarji, I have met with him over there in Philadelphia. That is where I was working. And one of my collaborators from there, actually my mentor's lab, they are working on pancreatic cancer and MYC protein. So they were the one who shown that triptolide actually works on pancreatic cancer through MYC protein. So I said, okay, this is something really solid, background to start my project on, gallbladder cancer project on. So I started working on it, and see in this, it's a very simple experiment. There are two cell lines that we obtained. I actually obtained these cell lines from my Chilean collaborator. So like I said, it is very hard to work on gallbladder cancer in America because nobody works on it. There is no funding for it because it's not a big deal over here. They will not give you funding because it is not a big deal here. So, and there are no cell lines available in America for this gallbladder cancer. So I try to collaborate with a lot of people in India. The problem is that they have plenty of cancers, but they don't really have cell lines. Somehow it's not very difficult to make cell line, but people just don't have interest in India somehow. So I collaborated with one of the guy in Chile who has developed these cell lines, and I obtained from them. Then it becomes very simple. So I tested this triptolide, and you could see, like, at increasing dose like 25 nanomolar, 50 nanomolar, in 100, even at 25 nanomolar, you see that viability of this cell line GBD1 and NOZ. There are two different cell line, two different genetic type actually. I would not go in the detail, but they have different meaning. Their genetic composition is different, but overall in both types of the cell line, it was found that it was very effective even at 25 nanomolar, which is really good, concentration. And Dr. Rakeshwar ji will tell, because he's a more of a drug person, he will tell that it's a pretty good dose, not very toxic, not very high concentration to start with. So it was very effective in gallbladder cancer cell lines. And I also tried several other cancer cell lines. Anyway, here if you could see, anti-apoptotic proteins like, CI, they are well-known proteins. People working in this field, apoptosis and cancer and other fields, they know these proteins very well. They are the markers for apoptosis. And apoptosis, for those who don't know about it, is something is a cellular procedure, cellular process through which most of the cells are destined to die after some time. So most of our cells die through apoptosis. But in cancer, that is why it becomes cancer, because apoptosis machinery go awry. Means those proteins don't, they stop, they make these cells to disobey the order of their death. It means that and these are the proteins which allow these cells to not die. So what happens, like when you use any anti-cancer drug, in this case triptolide, it should decrease the levels of anti-apoptotic protein, these proteins, and it shows very well here in dose dependent manner. Then also hold on. So triptolide also induces apoptotic cell death. So when you have decrease in anti-apoptotic proteins, it means that apoptotic machinery should work better. And it means that there should be more apoptosis. So as you could see in both cell lines, with the increasing dose, you see more apoptosis. Means these cells are, these proteins, this drug triptolide is killing these cells very well, even in dose dependent manner. Menilide, I'm using this another word because when you use animal model, here we have used animal model. So it is, you can also use triptolide, but this is why menilide was made, because it is water soluble. So it is something that it is something that you can go ahead. So it is something that you can directly use in the patients. So menilide and triptolide, don't get confused. They are same thing basically. For those who don't
go back on the slide, please.
Okay.
Do you think like at 100 nanomolar caspase 3 activity is not at all affected?
In where?
NOZ line.
Oh yeah, yeah. So you mean NOZ here?
Yeah.
Yeah. So, but this is what it is. So maybe they are. We don't know at this moment what's going on in this thing. But this is why I'm showing two different cell lines, which are representative of two different, genetic backgrounds. So I don't really have slide right here, and we are working on it. We are trying to actually sequence the some of the oncogenes, for example, KRAS, P53, especially P53 in these proteins, so that we could know that what is causing this particular cell line NOZ. I don't have pictures over here, but we have seen also that they go through something called NOIKIS, means they start after a few days, especially at day 2, they start floating on the cell culture medium. So they might be going through some of the mechanism that we are not aware of at this point. So very good point that you have raised over here. And since these cell lines are not very well characterized, and this is one of the big challenge in the field, it is an area that I am trying to explore further, that what exactly is causing. And this is why it is important to test as many cell lines as possible, because that also explains why some of the patients get resistant to some of the treatment. And this is not uncommon. I'm sure that there would be a lot of, if we ever reach to that stage where we would start clinical trial, I'm sure that we will also find some of the patients with gallbladder cancer that would not respond to triptolide. But so this is why I'm sure. But I had actually it's yeah. Somehow, sorry, I'm having a little trouble in listening to. So maybe you could write it down because it's easier to read it, or at the end, so you could write it down. So, but I'm not able to listen to it. But anyway, very good point, Dr. Chavra. So, like I said, when you treat these, nude mice xenografted with, one of the cell line, I'm showing one data, but we had other cell lines too, pretty much same effect. You could see that, they survived better, the one who were treated with Menelite. And you could see over here, so the tumor volume was, as you could see, actually the upper, I did not write it down over here, but this is, uh, you know, PBS treated, animals, and their, tumors extracted from them. And some of the representative, some of them are not really very well done, but some of them are very well done. So I'm just showing representative, pictures of the tumors over here. But they were, if you summarize everything over here, you could see that, GBD xenografted tumor volumes are pretty, low when they were treated. And 0.21 was the volume, mg per kg concentration. The dose was pretty effective. We also did a couple other doses, but this was very effective. That also corresponded with, in cell lines, that corresponded with almost 50 nanomolar. So that was very well tolerated dose also in these animals. And tumor volumes and tumor weights both were significantly reduced. Like you pointed out last time, in GBD, in tumor, this one is NOZ. So it's the same cell line that was not really giving very well, response. Here you could see that it's showing same effect. So you need more, double dose of 0.21 in this case. So it's basically one of the cell line is resistance. So it is very good for future studies. Why exactly this resistance is causing? What is the genetic makeup of these cell lines? So anyway, triptolide increases expression of pro-apoptotic protein cleaved caspase 3 in gallbladder tumors, grafted in nude mice. So I would just go to MYC here. Why exactly MYC is so important and why it should be one of the target here. So MYC aurora kinase protein represents an actionable drug target in P53 altered liver cancer. So like I said before, people have been trying in the field, pharmacologists especially, to develop direct inhibitor for MYC. And they have been, not really very successful to found, because MYC is such a protein that is very hard to target. The pockets, the pockets where drugs usually go fit in is usually not very labile, so it's not available most of the time. So what they have done here in this particular paper, if you look for it, they have tried to target the p53 MYC. So MYC and aurora kinase signaling cascade has been, they have been trying, and especially different types of drugs, families of the drugs, which try to indirectly target this cascade, not directly. Like I said, directly it cannot be targeted. So there has been some success, especially like I said, in this cancer, liver cancer, where p53 was altered. And MYC, like I said, one of my collaborators from Philadelphia, they also have shown that triptolide can target PDAC, or pancreatic ductal adenocarcinoma, especially those 20% of the PDAC patients which have MYC amplification. They give very good response to this therapy, this triptolide thing. So like I said from the previous paper, aurora kinase and MYC interact physically. And how exactly, when they interact physically, there are a lot of the drugs, not a lot, lot is a bad word, but there are, at least several drugs which target aurora kinase. And some of them are also in clinical trials. So this is how they have been trying to indirectly target MYC, because they interact physically. So if you use aurora kinase, not always, but in certain cell types, and depending upon the cell cycle, because aurora kinase is also cell cycle protein, it can work, and people have been successful, like I said before, in p53 altered liver cancer. So that was one of the inspiration for me, and I tried it here, and you could see over here, c-MYC and aurora kinase both in not only in one cell type, actually in both cell type, GBD1 and NOZ, both were pretty much responsive against triptolide when it comes to the mRNA levels of c-MYC protein and aurora kinase protein. Triptolide negatively regulates MYC and aurora kinase protein levels. So like I said before in the previous slide, mRNA levels were pretty down regulated, and MYC here were significantly down regulated with increasing drug dose, as well as aurora kinase protein levels. And aurora kinase, the phospho-aurora means activated form of the aurora protein. It's a kinase protein, was could be seen at higher dose, like at 100 nanomolar triptolide. So it was very significant. So this was one of the mechanisms. So like I said, triptolide is aurora kinase is one of the cell cycle protein that is basically marker for dividing cells, like you could see over here, these cells. So when you use triptolide it's a higher dose I'm showing because I needed to take the pictures, but that also works at lower dose, as low as triptolide 50 nanomolar. It doesn't work on 25, but at 50 nanomolar it works, and it is just 24 hours image, so you could see. But if you use like 50 nanomolar for longer time period, for example 48 hours and 72 hours, then you could see abrogation of these mitotic structures. So these mitotic structures are pretty much gone, means cells stop dividing if you use triptolide, and which is what we want for treatment of the cancer. If cells stop dividing, means they can be easily asked to go for for the death, for their apoptosis. So it is a good thing, and that's what we confirmed over here. So what this slide shows, that use of triptolide can stop their mitosis, means cell division will stop, and then they become more vulnerable for the treatment. So whole idea here is, if you use triptolide, we did not know at this point that whether it is aurora kinase or c-MYC protein, which one is being affected, what does it bind to. So that was not unclear. Still, it's not clear, but that's one of the question. And we know from and this is one of the signaling protein that I also work on, and I have been working for quite some time. Aurora kinase. Okay, so somebody raised hand. We will answer. Can you allow them to speak? I can't really now. sorry. Whosoever wants to ask question, would you allow them or should we wait until the end?
Dr. Singh, we can have a question answer session after talk. After the talk, we can have this.
Okay, sure. So, like, oh, sorry. So in this case, like you could say, I said, we did not know and we don't know yet. Triptolide, whether works on directly with, it's a more about mechanistic, thing. It's not, for those who are not interested in signaling and all that, it's not important. But as a researcher, we would also like to know that what are the targets for this drug here. In this context, we know from pancreatic cancer that triptolide works on c-Myc amplified proteins, but it could be something else in this context. In the context of gallbladder, it could be c-Myc or it could be something else that interact with c-Myc protein. We don't know yet. And we also know from my own experience, and other people in the field have shown, that aurora kinase cell cycle protein that directly interacts with adapter protein called NED9. And NED9 is actually one of the adapter protein that allows two oncogenes called FAC and SARC to come close and phosphorylate each other and make them more activated, so that they can facilitate the oncogenic signaling in the cancer cells. So all these proteins are important in the terms of cancer signaling. Anyway, so what little more about NED9 protein. This is one of the adapter protein that has many important domains when it comes to interacting with other proteins. For example, like I said, SARC family kinases, which are important, FAC and CRAC, aurora kinase, so it interacts with aurora kinase. So a lot of proteins which are relevant to GRAB2 is one of the proteins that is important for signal transduction of the KRAS signaling. So a lot of proteins interact with NED9, which interacts with aurora kinase. So we wanted to know that how exactly this drug is playing a role. So whether it interacts, so like you could see over here, NED9 is something that takes integrin signaling. TGF beta also impacts on NED9, and NED9 in turn interacts with SHIC and GRAB2 and couple other proteins and affects multiple canonical signaling pathways and ultimately NF-kappa B, AKT, ERK, and whatnot. So it was important to explore whether or not NED9 levels are also affected. So I would not skip this slide. So if you could see over here, NED9, NED9, FAC, and SARC, these all mRNA levels were affected by triptolide. So triptolide, we don't know yet at this stage. It's not published paper, and we are still working on it. But it's a totally early stage project where we don't know what exactly is going on, what exactly is the protein that directly binds to to triptolide. But it was important to look for what are the proteins that could be affected. So these are the bunch of other cancer related proteins which are also affected in the context of gallbladder cancer. Anyway, and I'm just showing there protein levels, which is pretty much standard. You look for mRNA levels and protein levels. What else it does? So triptolide treatment deregulates focal adhesion structures. So for those who don't know about focal adhesion structures are very important structure for cancer, not only just for cancer cells, other cells as well, but particularly in the context of cancer biology. These are these structures. If you look at the shining green dots at the edge of the cells, all these things, these are called focal adhesion structures, and they are very dynamic structures. So they are very important in order for a cell to move from one place to other. So basically they are marker or surrogate marker for metastasis. So one, for a cell to move from its origin of the cancer to other parts, this process is very important. So these structures are dynamic structures. They have to first form, then deform, and then form and deform, so that they keep moving from one place to other through matrix proteins, ECM, extracellular matrices. So this is one of the surrogate experiment where you could test whether or not focal adhesion structures are deregulated by triptolide. Because what usually happens in cancers, these structures, they become very strong, and more stronger they are means cells are more metastatic. But in this case, if you could look at it, triptolide at day one, it's a little higher dose because you need a better picture, but we have a lot more pictures where we have shown dose dependent manner effect as well. So in this case, you could see these are three representative pictures of DMSO treated cells, and these are the three representative pictures of triptolide treated cells. And you see these structures, focal adhesion structures are gone, pretty much gone. So it means that it's a weakening the cancer, the metastatic ability of the gallbladder cells. Anyway, it's another, I would not really take too much time. And then again, aurora kinase. Why aurora kinase is so important? Because it's found in so many other cancers. Most of the time, so in gastrointestinal cancer, it is very well amplified protein, mutated actually, mutated and amplified both. So again, I'll not go in the detail. Triptolide also reduces, EI4, which is, one of the factor for translation, EIF4, eMRNA levels. However, protein levels are not so much affected. Why? That we don't know yet. Because like I said, it's still, a very early stage, from project. So, but mRNA levels are affected by triptolide, which means that translation of the translation machinery of the cells are affected by use of the triptolide. So coming back to the, some testing, what exactly happens when you knock down c-Myc by siRNA? So we wanted to establish the effect, what you call it, interaction between c-Myc and aurora kinase. We know from other studies, c-Myc and aurora, they interact. But in gallbladder cancer cell, may not necessarily they interact. So we wanted to just confirm here, and we could see, when you knock down c-Myc, like here, aurora kinase levels go down. So it means that aurora kinase levels going down or depleted aurora kinase level by triptolide could be because of their interaction, personal interaction or physical interaction between these two proteins. This is what just we wanted to confirm here. I would not really go in the detail here because it's taking but so what we wanted to confirm whether MYC is one of the direct protein being affected by triptolide. So answer, you could see MYC, triptolide decreases MYC activity on GBC cells using MYC reporter assays. So we did MYC reporter assay and we could see that yes, it does. However, we are still working whether or not they are directly interacting or not, and it's not my area of expertise. So I'm trying to collaborate with some of the biophysicists, like SPR analysis, and those are to be done in future. What exactly triptolide is working on. So like you could see in this diagram, we don't know yet what exactly it is working on, whether it depletes c-Myc levels or it depletes aurora kinase, and through aurora kinase depletes MYC, we don't know yet. But overall, all these proteins are, what is the primary target, we don't know, and we will know only after SPR or some other analysis. And some of you who might be more familiar with those things may think from there and collaborate with us or whatever we want to take it. But these are the things that could be taken further. However, we confirmed that triptolide has no direct inhibitory effect on aurora kinase. So it was a in vitro analysis. So it doesn't really affect aurora kinase. So most likely, like I said, triptolide is working on c-Myc, but how exactly? Not through aurora kinase, because we just saw that aurora kinase activity is not in vitro analysis doesn't show any inhibitory effect of triptolide on aurora kinase. So it is something else that we don't know yet. So triptolide reduces, what it does, triptolide reduces the stability of aurora kinase c-Myc interaction in an overexpression system. So it was, co-IP study, and we shown that interaction is being, depleted by this compound. Anyway, so I would probably, because it's a little complicated here, what we found out, because like I showed before, one of the direct interactor of aurora kinase is NED9 protein, and NED9 protein is also affected by triptolide. So idea here is NED9 is upstream regulator of NED9 is TGF beta. So what we wanted to test whether or not triptolide is affecting NED9 through TGF beta, and yes, we saw that TGF beta mRNA was downregulated. That was the latest work we did, like just couple of, in March, before everything stopped here and everywhere else in the world. So at present, a lot of things to be explored further. What we know for sure that it is one of the drug that certainly kills most of the cancer, most of the cell lines that I have, and I have 8 cell lines. And I have done this analysis, almost all of these assays on 6 cell lines, except no NOZ cell line, which is one of the tough cell line to kill by this drug. 5 different cell lines coming from different patients, derived from different patients, are very well killed in vitro, like in cell lines, as well as xenografted model, means the tumor models derived from xenograft of these cell lines, the skid mice and nude mice, they are very well responsive to the drug. How exactly that is occurring, or the mechanism of action, that is not known very well. But at this point, whatever limited evidence we have here, we think, and I think particularly, and I might be wrong, that you can explore further, and some of you who are more experienced in these things can collaborate in future. But at this moment, what we think that probably triptolide is interacting on TGF beta directly, and we have not tested it. But we have other papers from other groups where they have shown that TGF beta receptor as well as TGF beta, both, and both of the receptors actually, both in different other disease contexts, have been down-regulated by triptolide in Chinese studies and in other studies too. So what we believe that TGF beta could be the primary target of the triptolide, and since TGF beta acts on NED9, which is adapter protein, which has direct physical partner as aurora kinase during the interphase of the cell cycle. Maybe since they interact closely, it is possible that aurora kinase, which also interacts with c-MYC in nucleus during mitosis, maybe that's how it is affecting the amplification of c-MYC, and that's how it is likely that the patients who have c-MYC amplification are affected. So like I said, c-MYC is very hard target to protein to target. There is no direct physical chemical that has been made so far. I mean, there are many, but no clinically approved compound which directly targets c-MYC so far, because they say it is very hard protein to target. But I believe that this is one of the mechanism for triptolide, especially in the context of gallbladder cancer. We have to still prove in pancreatic cancer, although they have different mechanism over there, my collaborator who work on pancreatic cancer. But at least in this context, what we believe that c-MYC is being targeted indirectly by triptolide. We have to prove that too. So yeah, so this is pretty much it. So now I would like to request you to ask your question, and then we can discuss. Hello.
So, Dr. Mahendra Singh, Rakesh here.
Yes. Hi.
Namaste. So great to hear. Yeah, great to hear you. And an excellent talk. I appreciate. Right. So I had a couple of queries and questions. This
Dr. Rakesh, I sorry to interrupt you. Actually, many panelists joined late, so I would like to introduce Dr. Mahendra Singh again. Introduction. Dr. Mahendra Singh, Assistant Professor in Department of Surgery, University of Miami Miller School of Medicine. He is a specialist in cancer research, drug development, preclinical studies, signal transduction, cell biology, stem cell and human genetics. Thank you, Dr. Mahen. You explained the difficult part of the talk in such a lucid and easy way that even I could understand properly. Okay, Dr. Rakesh, we can proceed with the question and answer session.
Sure, Rakesh, thank you. So I was just wondering, this triptolide, disodium phosphate, it's a water-soluble, new chemical entity. So if somebody has filed IND around it, or still it is
Yeah,
investigation?
so triptolide is basically, like I said, it is one of the drug that is available from thousands of
Chinese.
years, and people have been
Right.
working on it. But my collaborator, Dr. Ashok Saluja, he knew it, and he's a very smart guy. He's very senior person, so he knew it. So that's why the first thing he did, that he made the water soluble compound of it with the help of a chemist. He's himself a cancer researcher, cell biologist. So he made a compound called minilide. So this is why I have been using two different words. So triptolide is a generic thing. You can just buy from anywhere and you can work on it, but you have no patent on it. So anybody can work it, but there is no drug. Like people like you will not have any interest because it's not patent protected. So what this guy did, Ashok Saluja, he developed water soluble compound of it by adding some phosphate groups. You know it better, and so that it become and so he got the patent. So University of Minnesota, that is where he was working before, and that's when they did this discovery. I mean, well, you can call it discovery, but this was the patent which was held by University of Minnesota, and in the name of Dr. Ashok Saluja. So they have the patent for this. So it is IND in a way. It is IND. But pretty much both compounds, like the triptolide also has same signaling and everything same. That it's just phosphate group addition, and that makes it water soluble, and that also makes it like patent protected. Otherwise both of them have similar profile, pharmacologic profile, and everything is the same.
So seems like this is a pro drug, and because triptolite itself is active, and when you make it derived with disodium phosphate, it becomes more permeable and soluble,
Yes.
and so gets to the site quite effectively. So I'm
Yes.
just looking at the structure. It has got like steroidal core, right? If
Okay.
you look at A, B, C, D, zinc,
Yeah.
and you know there are a couple of medicines called abiraterone and all kind of things. They are being used for prostate cancer.
Mm-hmm.
Okay,
Mm-hmm.
so this structure has a high degree of similarity with that. You
Okay.
can think of repositioning or repurposing this for understanding how you can make use of it for prostate cancer. This is point number one. Second thing is, recently we have seen the dexamethasone. It's a
Yeah, that's
corticosteroid.
yeah.
So it has also similar structure. So
Okay.
and what happens in now in current pandemic, right?
Yes.
Look at it, if there is certain role of aurora kinase to kind of regulate or deregulate certain genetic machinery of RNA, which is there in COVID. So just have a look. My sense is it might be useful to repurpose this your molecule.
Sure. Yeah, that's a good idea. I'll see. I'll see.
But more importantly, look for this prostate cancer. I think this has high degree of possibility to find some application there.
Yeah, so that's the thing. I mean, like in academia, anywhere, it is very hard. If I look for prostate cancer, then there will be no funding for me. Even for gallbladder, I have a track record, but I'm having hard time to get a funding. So it is very hard to keep changing your area of interest. Like it works in US. I don't know. It might be a little easier in India.
So you might find some collaboration, because I tell you,
sure.
for prostate cancer, not many medicines are there. This is only one medicine that works quite effectively. It's called abiraterone. So, and it's very difficult to make because it comes from soybean or pig's liver. The starting material gets isolated from there.
Yeah.
So, and then you need to have a containment facility to manufacture that medicine because of the steroidal nature.
Sure.
And triptolide will not have exact harmonogenic property, but it will have a kind of a mimic core structure of steroids. It might help you to design around and find some more applications.
Sure. Yeah, sounds good. Thank you. Thank you. I'll think about it.
Mahendra, very nice talk.
Yes.
So
Thank
if I may ask, like, what's the status of immune-based therapies in gallbladder cancer and even pancreatic cancer?
So immune-based, so gallbladder cancer, there are no therapies. Nobody is, working on it. It is
Why
one of
not?
the, this is why you guys are in India. So this is why my purpose was of this talk is to encourage you people, because to be honest, I may not be able to continue with this kind of cancer working in here. Everybody is discouraging me. Oh, this is something, nobody, there are no patients here in America. So for you, maybe I'm just, suggesting, think of it, because these are the opportunities. Indian people just don't think about that. They are running behind something that westerns are already working on, like breast cancer. You don't need to work on breast cancer, because breast cancer these people are working on anyway. So coming back to your question, no, there are no trials on any immune related therapies on gallbladder cancer. In pancreatic, yes, there are, because pancreatic cancer has lately picked up very well in America. In last 5-6 years it has picked up attention. A lot of new uh, therapies are coming up, and people, new grants, and a lot of activity. So they have been trying to do clinical trials, especially using immunotherapies, a lot of immunotherapies. But unfortunately, CAR T and those things are not very successful in pancreatic cancer, because for immunotherapy, so they say, I'm not immunologist, so I might be wrong, but they divide cancers in two types of cancer: cold cancer and hot cancer. So pancreatic cancer is considered cold cancer in the terms of immune cells. So when you use these PD-1, PD-L1, and those kind of therapies, they are trying. They haven't really got much success so far. Like early trends from the different trials, so a lot of combinations with uh, available therapies, chemotherapies plus or targeted therapies plus PD-1 or PD-L1, but they are not very responsive. Yeah. In pancreatic cancer, there is a lot of work going on, but in gallbladder, whole field is empty. Nobody is working on it. And I know a lot of Indian people too, but unfortunately in India, it is basically doctors who are working on gallbladder cancer, and like they don't have time or proper understanding of these things.
I think
So
Subhash Chauhan is working on the pancreatic cancer, right?
yeah, he's working on
Yes.
pancreatic. I am in touch with him, so not recently, but when he himself told me that, what, if you have to live in America, forget about it. So this my main purpose is, no. So we all survive, right? We have to survive also. So, but gallbladder cancer is very close to my heart. I was the first person who did PhD on gallbladder cancer in India in 2003. So, and it is still very close to me. So I really want, whole purpose of my this talk is to like inspire people in India, like you are in
Yeah.
position, a big
Yeah.
position. So
I mean,
think of it.
yeah,
It
I've
never
been back
happened.
here 2 years only, so I'm
I
just still making
yeah,
roads. So like, it's not easy here also, but like we
I
are
know
all
it's not
trying
easy.
to make ways. Like,
I know they have different, but in future, like think in those terms, and
right.
and then we can collaborate. Even if I'm not working on it, future we can.
Sure.
I have plenty of ideas, but here they
Sure.
would not give me grant, because I'm
Sure.
not that big shot, right? If I was, maybe they would give me. So I have to prove my things, and then by then. So yeah, but this is an open area for India, and now they are opening up new institutes, and new grants, and all those things are there.
We got some
And
collaborators for samples and everything, so I'll get back to you.
yeah, no,
Uh,
sure. But
Can
thank
I
you.
yeah,
make a comment
yeah,
here?
Dr. Mahendra.
Yes, sir.
Dr.
Yeah,
Mahendra, I
yeah.
had one question. You in your initial slides, you said there is high prevalence of gallbladder cancer in pancreatic vein. What is
Yes.
the reason behind it? It is because of some eating habit or some genetic predisposition. What would be the possible cause? Is there any
Yeah,
study?
very good question. So when I was working back in those days in India, I used to really interact with almost everyone, all the doctors, surgeons, and everything. And it was a big thing, big discussion. So there is a group back in Varanasi who he's a surgeon called VK Shukla, and he now I think I heard that he is head of the IMS BHU, like the medical center. So this guy was the one, and I had met him a couple of times. He did some early studies in like in 90s, uh, where they had shown some of the heavy metals and all that. But there was another study, so they showed that how exactly some heavy metals I mean, I don't remember exactly which ones, but they have multiple papers. One of the paper actually came in 97. That was published in BMJ back in those days. So that was a study to look after. So it was heavy metal, and there was another unpublished study, but I know a guy from Patna. I don't remember the name, but he told me long time back that they had shown that this particular thing called mustard oil. So you know that North Indians are the only one people who use mustard oil as the primary, uh, oil for their food. So mustard oil is itself not bad, but somehow to bring the jhas or the pungent, pungent smell, they contaminate it with some kind of thing. I'm forgetting the name, but it's a chemical that has been in the talk for a long time in India. That what they exactly add in the this is some I'm forgetting the name for it, but it's a flower. I know it's a Hindi name.
Arjimon.
Argimon, exactly. So the argimon contamination with mustard oil was something that they were thinking, and they had some preliminary finding also. And this guy was working. But I will remember his name later on. So that makes more sense because this is the only part of India, Gangetic Belt, where people use mustard oil, and mustard oil is contaminated with argimon. So that could be another thing. But these are the here and there, little bit studies, but no solid evidence so far. But it's true that if you talk to any doctor from South India, they would say that I used to work with doctors in SGPGI, so they used to tell me, especially the residents coming from south part, they would say that I have seen gallbladder cancer only in my books. I have never seen any patient. It was the Lucknow, it was the Delhi, it was the Patna, Kolkata. That's where they find the patient, and plenty of the patients. So something to do with, and genetic, yes, genetic could be some, but genetics is so complicated that to pinpoint, especially in cancer, no one gene is responsible. So I was the first person who shown 35% of the KRAS mutation, and then one of the polymorphic variant. But after me, there's so many people from the same area, they showed, oh, this polymorphism, that polymorphism, but that doesn't really give any clear cut answer.
Thank you, Dr. Mayan.
Yes.
There are few questions from the participants.
Sure, please.
There is one, Tanupal is asking, any other medical treatment to cure GBC other than operative measures?
Not exactly. That's the unfortunate part of it. So basically, what happens when they report to the, so okay, let's look at the anatomy of the patient. So what happens, like, since gallbladder is embedded in almost to the liver, by the time the patient report to the doctor, the disease is spread to the liver already. So it is very hard. But let me tell you one thing. In America or other countries like advanced countries, gallbladder cancer is also not a big problem because people are like more what do you call it, I mean, they are more careful about their own thing, right? Back in India, we don't really go to the doctor until it becomes really very necessary. In America or in Western countries, people are like, they cannot tolerate small pain, so they go to the doctor. One of the good thing that if found early, if detected early, it is very easy to treat because all you have to do is take out the gallbladder, and gallbladder is one of the organ that if you take it out, it doesn't cause much problem because gallbladder can be, yeah. So in America, this is another reason it is not a big deal because people who, first thing, that it's not very common, and even uncommon one they report very early. But back in India, by the time they report, it's already spread. And so, like I said, surgery is the only cure and very good cure. But if surgery cannot be done, then most of the time nothing can be done. They give them chemotherapy, but chemotherapy is only to alleviate their pain. There is no targeted therapy. Like in breast cancer, we have a HER2 receptor. We know that HER2 receptor and a couple other receptors are expressed, so people have made drugs against it, right? Like tamoxifen and whatnot. So those are called targets. But in gallbladder cancer, targets are not known.
Okay, another question is, which genes, it is from Abhishek Kumar Jaiswal, which gene would be most important gene for targeting GBC?
Yeah, that's a very good question. Yeah, very good question. So thing is, there is no clear answer to it. So like I said, in my second or third slide, there was a two big papers. So most of the time there are small papers because some papers are from India, some from Chile. Japan is another country where they have a lot of this cancer. But in Japan, the problem is like etiology is different, means the cause for the cancer is different. So they have different pathogenesis. So that's not relevant to us. So in Chile, in India, there are small papers, nobody cares about them. Two big studies so far came in, one in Nature. Actually, both of them in Nature, one in Nature Genetics and another one was somewhere else in main Nature, maybe 2014 and 17 or 16. One from China, one from, these were really good studies where they did like hardcore genome sequencing and everything. And they found that ERB2 and ERB3 were the most prominent genes in their patient. But it may not be true for Indian patients, because our patients may have different genetic makeup. We don't know. So unless we do some, and I think there are some studies, one collaborative studies done by Tata Memorial Cancer Center and one of the guy at MD Anderson here in Houston. They also shown one of the ERB2 protein, being mutated and amplified in gallbladder cancer. But that is only until 30%. So there is no, like I said, there is no clear answer. What we have, and this is what we call it personalized therapy. There could not be just one gene which is important for all cancer, all patients of the gallbladder cancer. Some patients could be affected, could be important in the sense that they have amplified MYC gene, and some of the patients may have amplified or mutated ERB2 genes. So depending upon what patient may carry which kind of mutation, they need to be targeted accordingly. So this is what is precision medicine, and that is where the whole world is going. In America, in Western, and even in India, in big centers, they always go for some kind of sequencing. Not for all cancers, but some cancers where definitions are well known, like breast cancer and all that, they go for these mutations: BRCA mutation, BRCA1, BRCA2. So in gallbladder cancer, it's only these two genes, MYC, I would say MYC and ERB2 family genes. ERB2 and ERB3, which could be important depending upon which patient we are talking about.
So, do we have time for one quick remark?
Sure.
Okay, so
Yeah.
the first thing, I just want to tell few things. So we have very nice group in JNU where we are addressing heterogeneity of head neck cancer by using multi-omics and system medicine. So
Okay.
I would say that now that is not the situation that people are not working on modern technology in India. And we have this big group funded by DPRP, and we are around 12 people coming from different School of Computer Science, Medicine, and Biotechnology, Life Sciences. So we will be happy to collaborate with you and also some of your partner in gallbladder cancer over here. So we can really tie up for that kind of thing, and then you can combine your drug to this basically study and other things. Okay, and
That's
this
very nice. Thank you.
and the thing which you were telling in the molecule, but this is a very common target. So how your drug molecule is able to differentiate with the common target versus the cancer specific one? But probably
Which,
this is the last drug which you were talking. But probably this is known in the literature. I am ignorant, so we'll discuss later. But I just want to say that feel free to get into touch with this group, and it's a very nice group. We are funded for next 5 years, and
that's great. So are you in that group?
I am one of the member for proteomics group. So I deal with the
Oh,
proteomics, and then we have a
that's
transcriptomics, we have system biology, we have epigenetic group. So and we
that's very
have
nice.
a lot of computer person into it, and then soon we are in a process to write, or you can say we are going to get a grant from ICMR, and that is about systems medicine. And there we are thinking about two cancer, one head and neck cancer, and another basically gallbladder can also be taken, because this is a India centric problem, and especially
Exactly.
when the people are having H pylori problem. And
Mhm.
Rupesh Chaturvedi is also working with us in this group, and so
Okay.
he is basically looking at H pylori induced gastric
Yeah.
cancer. So
Okay.
probably it will be a nice to have you in this basically group and discuss things together.
Sure. Yeah,
So
we will be in touch. Thank you, sir. Thank
okay,
you. Yeah.
yeah, you are
We
most
should do
welcome.
something for India.
Yeah, okay.
Okay, thank you, Dr. Mahan.
Thank you very much.
We have had a very good discussion and very nice talk. Thank you for joining us from Miami.
So thank you for inviting me to. It's
Yeah,
a
we
great
hope to look
to interact.
forward. In another occasion, we will definitely contact you.
Sure.
Thank you very much.
Thanks again.
Our next speaker, Shiv Kumar Misraji, is due to some reason is not able to join. So I call upon our next speaker, Dr. Sovi Valeri. Dr. Sovi. Hello.
Hello, can you hear me? Good morning.
Yeah, Dr. Sovi.
Yes, yes.
Thank you for joining us. You have been with us since today. Dr. Sovi, I would like to introduce him. He is scientist E in DBT, Ramlinga Swami Fellow. He is in ICMR, National Institute of Medicine, Tarnaka, Hyderabad. Dr. Sovi is PhD from University of Eisenbach, Germany. He has been fellow DBT Ramlinga Swami, SIR, NIST, associated with NIH Research Fellowship, was visiting fellow in NIH, USA. Also have held a post of scientist B in DIPAS, DRDO, Delhi. Dr. Sovi has a very good academic and academic record. He has got NIH Merit Award. 2014, his research on CC2D2A was one of the highlights of NIH research. As far as publications are concerned, he has numerous publications in international journals of repute. He has been a speaker on numerous international and national conferences and seminars. Has more than 19 publications. So I feel proud to invite Dr. Sovi to present his talk. Welcome, Dr. Sovi.
Thank you very much. Can you see my slides? I'm sharing my slides.
This one is not visible right now, but you can share. Well,
Okay. Anyway, thank you very much for the kind invitation to participate in this international conference held at Kamala Nehru Institute. I'm indeed very happy to connect with my old friend, Dr. Rakesh Bandikar. And I'm also happy to be part of your team, Professor Pandey. Thank you very much. And it will be very good if you can let me share my slide.
yeah,
No,
it is visible.
it is visible.
Yeah,
Okay.
very good.
So today's my topic is about the genetics of body plan architecture, health, and disease. By way of training, I have worked extensively in the field of genetics since my PhD. And up until now, I am still working on human genetic diseases and trying to work out its basis. So, okay, what is let me go to my mode. Hello.
Yeah,
The slide is changing.
it is not sliding.
Okay. Okay. Let me. Hello.
Yeah.
So I'm trying to connect it from my alternative phone screen. Please allow me to share that slide from there.
Another.
Okay.
You are connected from another device or?
One second. I am trying to connect from my mobile another PowerPoint, so it may work faster. You can see on the screen, either one will be working. I am going to switch off the one current one.
Okay. Okay.
So let me. So can you see a slide now?
Same one.
The same
Earlier
slide.
one is.
Another one.
Hey, if you move it, then it will be.
Can you see a second slide now? Can you see? Hello.
Same
So your
is
second
on the screen.
profile is blank.
Profile is okay. On that, can you see now?
You have two profiles.
Yeah, the second one, please allow me to share the slide because that may be running faster.
No sharing. Correct.
Yeah, both are allowed.
Both are allowed. Okay. So now, can you see the first one again in one of them with the title Genetics of Body Plan? Let me again.
Video is not starting in here.
One second. Let me. If I am allowed, it will come out.
Second read.
Yeah.
And device you open the video.
Yeah, I opened the device. And just trying for. Yeah. Now I am sharing my screen. Yeah, during mobile sharing, video is first. Okay, video is first. Okay. So,
Not video.
no video. Okay, that's okay. But I can get my slide. Just a minute. It is starting. The slide is on. No.
Yeah.
Slide has come.
Yeah. You can.
Okay. So this is my first slide now. So as you all know that the secret codes for our life is encoded in the chromosomes. And why I'm showing these chromosomes are because the expression of the genes in these chromosomes really makes a big difference. Because what I'm trying to tell is that gene expression is everything we see in our world in form of life. And this is the big one is the X chromosome and the small one is the Y chromosome. And if the genes are expressed from these two X chromosomes, what you see is Kate Winslet, you all know the heroine. And if we are getting one X and one Y, that's a male. So that makes a big difference. This is the simple point of gene expression. With respect the codes you have in your chromosomes, what you see is quite remarkable and amazing. And this is because the genes are coded in DNA, nucleotides, and which is translated into RNA. Actually, every cell of our body or any organism codes basically the same DNA codes, but they are not expressed in every location. For example, rhodopsin is only expressed in the eye, not on the feet of your tail or on the at the nail of your feet. And once the RNA is expressed in the eye for rhodopsin, it will develop, or produce amino acids, and these amino acids will create the protein required for rhodopsin. Or for that respect, you can go for different organs or organoid wherever some special molecules will be produced. And in other part, there are constitutive genes. It will be expressed all over the body for basic purposes. So that is the basic of gene expression. The overview of my talk will cover, biological phenomena where I already told you that there's a blueprint for everything. It is not random events. And this blueprint is basically encoded in our current understanding in genes. And these genes form basic programs. And there are different types of programs. Some of them are linear. That means they will only go in one way. They cannot go back. And the second one is, non-linear, which is, it will be repeating again and again. So for example, the linear one is for the body structure. When we are developing our body, develop only once that way, most of the cases. And for the biological rhythm, it is repeating every day. Or for that matter, some of our metabolic functions repeat every day again and again. And some of the programs, these genes make the cogs and needles of our clockwork, something like that in a cell. If something is wrong in that network of molecules, you can get inborn errors, or you can also have diseases developing as your system is getting, wear and tear with the molecular function. With that, I start. So this slide I used to demonstrate the fact that there are multiple forms of life on Earth. Simple insect, starfish, and the majestic tiger, or anything what you can see on this Earth. Even though the many forms are visible, the very basic for developing all these forms of life is limited to a set of programs. And you can count them like, WIND program, Notch program, Hedgehog program, Gen, Stat, Map, etc., etc. These are the basic developmental programs. You can compare this something like, the Zoom program now we are using, or TikTok or WhatsApp, something like this. These are different modules used for a different purpose in your development or functioning of the body. I'm not going into the details of all of them, but I will touch upon some of these developmental pathways as I go, and I will show some of the classical examples from the literature as well as my own work. So I told you that there are programs for structural layout that is called architecture, and I call it a linear program. The best example is that we have a single ovum. When it is fertilized by a sperm, it will go systematic cell division and develop a highly complex body like human body. And this is a peculiar example of a linear genetic expression program. And it is integrated with the multiple modules I told previous slide. And the most important thing here is that when the human body is developing, or from a single cell, or for that matter, many metazoans, there are multiple binary decisions made how a cell should go forward and make its new fate. It is slightly connected with the stem cell, science as well. You may know the details. And here I try to demonstrate this phenomena using a single experiment. You know that we have a head, and our predecessor, the monkey, has a tail. And this is simply, a chance event during the early development. What should become a head, what should become a tail, or you can go for that matter an anterior and a posterior region. So this experiment is elegantly shown in planaria. All of you know that if we chop the planaria into multiple pieces, each piece can develop its own head and tail if everything is normal inside that organism. That means if all the genes are functioning perfectly all right, when you cut this planaria here into two pieces, the part missing head will make a head, and the part missing the tail make a tail. So that means there are programs already available. And this experiment was elegantly demonstrated by Alexandros lab, and they have dissected this planaria with RNA experiments. And what they have shown that in this control, if you cut this animal, that line, dotted line is showing, it will recreate a head and a tail properly. But if you do knockdown of this beta-catenin pathway, this is part of that Wnt signaling, what will happen that there will be no more tail. So that's why I put a title here that there must be beta-catenin working properly to have a tail. On the other hand, if you, along in that genetic program pathway, if you knock down the disheveled gene, what will happen is there will be no more head. It will be only tail. So it is very clear that there are specific genes for head and tail. And this pattern is applicable even to our human structure. Now I go to another simple example, which will be easy for all of us to connect. The structural layout of our hand is a quite unique design in the development of, the mammals, especially primates. And you know that we have a thumb which can interact with all other fingers, and this made a lot of advances for human race. And we have used, along with our developed brain, this hand made a lot of difference. But all of us normally have a very fixed position for thumb and the little finger. How this unique design repeats again and again, and the detailed analysis shows that during development, this is a mouse embryo now. We know that there is a sonic hedgehog. I'm only showing a part of that story. So this is in the picture, you can see the forelimb and hind limb. And in the hind limb at the embryonic stage around the E8, E10, there is a bud only. And that bud specifically expresses one side sonic hedgehog. And where the sonic hedgehog is expressing, that will be your little finger. And the thumb is always fixed. And if the expression goes wrong in this pattern, and this is applicable to both forelimb and hind limb, and if this expression goes wrong, your pattern of development of the finger is completely messed up. And this was elegantly shown by one of the scientists in NIH in 2008 only. The theory was already known very well, but they did systematic temporal, or what is it called, targeted knockdown of the gene in this limb, what they have done. And if you break down this gene expression early, you will miss many fingers. And as the time span changes, you get more and more normal. That means the early in the development, you block the expression of sonic hedgehog, you lose many fingers, as you see in this picture. So here is a timeline that, so on your right, you can see that at the early development of the embryonic day 9.5, if you knock down sonic hedgehog, you will miss almost all the fingers. And that is how you lose the pattern. And there are more complicated structures coming up if there is a mess up in the sonic hedgehog signaling. I will touch upon that with my own data later. And now I worked extensively on the eye field. As you may know that I was with the National Eye Institute, and my PhD was also in the photoreceptor area. And we know I worked with Drosophila, and for architecture of eye position, there are classical examples. And there is a transcription factor called ILS. And if this transcription factor is working properly, in Drosophila get this beautiful compound eye, and Drosophila also have additional eyes. But what you see in the picture is the main one is the compound eye. And if this eyeless mutant, the mutant is named after the gene. So when there is no eye, it is called eyeless. And when in this mutant, you can see that if the gene is not expressing, there is no eye. So again, the emphasis is that there are peculiar genes. Their expression is very peculiar, and it is localized and spatially controlled. If it is not happening there, the eye will not develop there. And scientists were smart enough to show that if you take that gene and express in the leg, or the picture on the right, you can take this transcription factor and drive expression with the help of a gene expressing in the leg. You can actually develop the eye on the leg. It was a very beautiful experiment done back in 1995. And again, I'm touching upon the eye itself. Inside eye also, there are so many binary decisions are happening along the way of development. And here, a classic example, you may all know that we have 95% of rods in our retina, and around 5% cones. And cones are also of subdivisions are there. But for the simplicity, I say rod and cone. The thin ones are the rods, and the conical big ones you are seeing are cones. And how these are determined. See, in the development, early development, now I'm showing data from the mouse, and it is my own postdoc Manders, Dr. Anand Swarup's data. And so our retina inherit early development, let's say around the E7 embryonic day, a set of precursor cells, and they are destined to become cone cells, as you see in the picture on the left. And if there is a transcription factor expressed in these blue-purple cells, which is called NRL, neural retina losing zipper, and those cells which are expressing NRL, they become cones. That's how it is happening. That is the binary decision. And here is the real data. If there is no NRL, there will be no photoreceptor, I mean the rod receptor. Because on the left, we have marked in the normal rods in a green antibody that specifically stands for rods. And when you have a NRL knockout animal, there is no rods and there is no staining on the right, and you can see largely it is missing in that picture. And similarly, we can also do so many experiments with the different sets of genes. This is a disease condition where I have used a knockdown experiment to kill a gene called BBS9, which is required for some aspects of the photoreceptor development. And if you use that gene knockdown experiment and we knock down BBS9 gene, and then what happens is the eye development is actually messed up. And you can see the details of this normal and the disease case. On the left, you can see the zebrafish retina is multi-layered properly. And in the diseased condition, we mimic the diseased condition in human. That the BBS is a Bardet-Biedl syndrome. I will touch upon on later. And if you knock down this gene, what happens is that the entire retina is messed up. It cannot function anymore, and the animal is blind. And now I will switch a little bit. And last 10, 15 years, we have realized that most of these modules I talked in the very beginning, that I said there are approximately 5 to 6 fundamental pathways for developing your body and for other functions in the body. And these programs are often seen capsulated in a structure called cilia. And yesterday, Dr. Sunil Kataria from JNU was talking a little bit about his photoreceptor in Chlamydomonas, and their research. The people from the Chlamydomonas area have done extensive fundamental research in the field of cilia, and now the clinical side is merging with the fundamental research coming from the cilia field. And actually, fundamentally, the cilia in Chlamydomonas and the cilia present in the human body are equivalent, and because their basic structures are same, so that we can model many human diseases in Chlamydomonas or ciliated cells analysis in vitro. And this is what actually I said. This is a cilia, which we call flagellum, but basically cilium and the flagella are fundamentally same structures, because both of them have a basal body, which is one of the mother centriole, and which forms a shaft that is called the cilium. And the cilia is actually present almost on all cells of human body. It is present in our airway, in the lungs, it is in our brain ventricle, it is in our reproductive system. And here you can see an embryonic node, and an embryo is on the left, and there's a dark spot that's a node, which is a structure present only for a few hours in human development. And it is present in the cilia, are present in our ear, are present in our kidney, bile duct, even in our retina, the last picture on the right. And if any of these, if you have a fundamental defect in a cilia development, it can affect multiple systems in your body. And these days, the defects due to cilia gene are called ciliopathy, because it will have multi-system disorders, and it is called syndromic. And there are multiple forms of syndromes. One of them is this Bardet-Biedl syndrome. And I told about one gene earlier. And in Bardet-Biedl syndrome, for example, you will have obesity, hypertension, retinitis pigmentosa, that is blindness, polydactyly. I was talking to you about the hand digit patterning. And if the cilia gene goes wrong, you will have sometimes multiple fingers on your hand. You can have cleft palate, open neural tube, many things like that. And this is another disease called Meckel syndrome. And when I was in NIH, I was working on this problem. And this was reported in 2008, a human fetus with Meckel syndrome. Meckel syndrome means it is embryonic lethal, and this fetus have open neural tube, as you see on the right, and on the hand you can see there are polydactyly. That means more than record number of 5 fingers on each hand. And at that time, they identified by GWAS, that is genome-wide analysis, association study analysis, they found that there's a gene called the CC2D2A, but its function, molecular function was not known. So I made a knockout of this gene in mouse. And just like in the human case, the knockout mouse is embryonic lethal. You can see on the left, there is an embryo at embryonic day 16 and an embryonic day 21 or 18 or 20, the animal will be born normal. And in this case, you can see that the phenotype is pleiotropic, and I have received one animal out of 1,000, which was surviving for some reason, and it had a hydrocephalus, the small one animal you can see there. It had this bumpy because the ventricle in the brain is not functioning well. The cilia is not there properly functioning. And similar situation is seen in ciliopathy patients with hydrocephalus. You can see a baby on the top there with the hydrocephalus. And a few of the animals from this knockout were showing polydactyly. You can see the normal with the required 5 number fingers, and the defective animal has an additional finger. And you know that one of our hero has a polydactyly. I don't know how many of you know that. Hrithik Roshan do have a polydactyly. And this some of our knockout animal, for I am talking about now the CC2 knockout animal, which also had situs inversus. That means even though you look bilaterally symmetrical from outside, inside your body, your internal viscera is not symmetric. Your heart is always pointing to the left, but in this animal, the heart is pointing to the opposite side, and it was also missing some of the internal viscera. So this problem suggested us that, yes, there is some problem with the embryonic node. So this is actually an embryonic node on the left, an EM, scanning EM. And you can see at the node, there is a small depression, and the depression in the insect shows have a lot of cilia. And if this depression, actually this depression is only present for 2 to 3 hours, even in human embryogenesis. And here the video is working now, that the Japanese experiment that shown that this cilia always beat towards the left, and then that creates a morphogenetic gradient of molecules present at that node. And because of that, what happens is that you will develop this internal asymmetry in the body. And if your node is not functioning properly, you will have, some people had joked earlier that somebody is missing a heart because it's not on the left. And there are instances like that, but there is no chance of living with that heart missing. But you can have the asymmetry, because there are asymmetry even in the number of lobes on the left and right of the lungs inside us. So I actually digged the embryos of my knockout animals, and here you can see the immunohistochemistry as well as EM of control versus knockout. In the control, there's a nice embryonic node, and which is having a lot of cilia. Whereas on the right side, you can see the embryonic node is not properly developed, and there is no cilia present. And this is the reason why you are having this internal asymmetry in that animal. Moreover, this animal's neural tube development was also abnormal, because as you see in the normal case, the neural tube do have nice cilia stained in green, and which is absent in the knockout. And if the neural tube is lacking some of the cilia, and cilia do have a lot of developmental pathways. I mean, the cilia host a lot of developmental pathways, and if they are not working properly, you can see here, I have talked about Sony Kajau earlier. A normal neural tube should look like this, where there is a neural plate, and there should be nice Sony Kajau signaling. But in the knockout, since there is no cilia, this Sony Kajau signaling is not working properly. So it is similar to the hand digit patterning I referred earlier. If the signaling is not working properly what happens is that the neural tube will not close properly. This is another marker from the same neural tube. And if some of these things are not working properly, you can have situations like this. This is called exencephaly. The neural tube is not working properly and it is not closed. And such cases that the fetus will be most probably lethal. And I have actually digged out the cause of this mutant. What happened when you knock out this gene? What happens in the cellular level? And we already analyzed the MAP cells from this knockout, and we found out that in the control animals, you have a nice basal body, and that is creating a cilia that is in green. But in the knockout on the right side, what happens is the basal body is still there, but it cannot develop a cilia. So we did the EM level analysis. We found that something is missing in that knockout, and we know that there is something called subdistal appendages, and you can see that which connects to some of the microtubules for getting cargoes into the cilia for its development as well as for function. Such structures are absent in the knockout. So we are suspected that something is wrong at that level. And we had actually many other ways to analyze that subdistal appendages. And the immunological studies, immunohistochemistry study indicated that the subdistal appendage is missing. That's why I actually did this EM analysis. And this is a realistic picture or a schematic showing that how a basal body with a subdistal appendage and it connects with microtubules in the cell. And that is, we have done that in much detail. So I took a monkey photoreceptor, which is a modified cilium, and you can see that there is a distal appendage and a subdistal appendage that is a small circle inside the circle, and that is we made an antibody against the CC2D2A antibody and the immunogold analysis, and that specifically went and attached to the subdistal appendage. That means what we said in theory is true because we can prove that, and we also did this in the kidney cell. Here again, that immunogold tagged antibodies for CC2D2 specifically binding to the subdistal appendages. So two-way we have proved that yes, indeed it is part of that. And we, I also want to tell that since that cilia is not present, and I said again in the previous slide that photoreceptors are modified cilia, and in the left on the bottom panel you can see that in wild type animals the retina patterning is proper, but on the right side the photoreceptor layer is completely messed up in the knockout because the cilia is not there. And this is why yesterday Professor Sunil Kataria from University of JNU was telling that cilia do develop blindness. This is one of the reasons. So something wrong goes inside the cilia. It can develop a congenital blindness, or you can have retinal degeneration. There are multiple ways to get this blindness. So based on my data, we have developed this model. For clarity, I am showing this picture to you. So there is a cilium, and the blue part is the basal body, and next to the basal body you can see subdistal appendages. There are 9 of them, and they are made up of multiple proteins called CC2D2 and many other things like inine, ODF2, etc., etc. And now I show you another example for non-linear gene expression, how it impacts. And we can call the best one is the biological rhythm. And you all know that we wake up morning and we sleep evening. So we are connected to the world with respect to the changes in the light pattern in the world. And it roughly has a 24-hour rhythm, and our biological rhythm is tuned to that. It is very important that a butterfly must know when to go to a flower for taking its nectar. If it is going in the wrong time, what happens is it will not get its food. So the purpose of biological clock is that it allows the organisms to anticipate and they follow a rhythm and a pattern. It improves their fidelity as well as their fitness in the biological needs. And the plant here I am showing is a sunflower. What it does is that it is tracking the movement of the sun, and when the sunset is happen, it knows that the sun will come again from the east, and it is reoriented. So it knows that it is anticipating that the daybreak will come from the other side. So the circadian clock is something that allows the organisms to, anticipate and pre-plan its, behavior. But these are old experiments. But those days, the genetics of this circadian clock was not clear. But then there was search further, and this is an experiment done by Browning in 1957. He have systematically analyzed whether the closing and opening of this plant days is rhythmic, and he proved. He's a German scientist. He went very technical and connected electrodes to the plant leaves and figured out that yes, there is a rhythm, and it is very close to the rhythm of the daylight. It changes in the earth. And furthermore, there is another German scientist who is called Jürgen Herschhoff, who is one of the pioneers of the circadian clock analysis in human. He did beautiful Bunker experiments in Munich. Me and Sunil was there, I think, at least. And he have found that the humans have an endogenous rhythm. Why it is called an endogenous rhythm? Because we have a rhythm, but what happens if the light goes? So you are in complete darkness. But even if you are kept in complete darkness, what happens is you can see that there is a LD means light dark, and then LL means continuous light. So your rhythm speed is little bit the pattern or the frequency of repeating is little bit delayed. But when you are put back into LD, you are synchronized to that 12 hour. And when you are put into a continuous darkness, what happens is that you will still wake up in a almost a 24-hour time. That means without any external signal from the environment, you will be able to wake up and you will be able to sleep roughly close to 24 hours. So these old experiments gave a lot of ideas about there is something genetic program inside, and we are talking about before the genetic era, and people had vague ideas about all these things. Then what they did is they created a concept that yes, there is something called a central oscillator, and the oscillator is getting some input from the external environment, which is mostly the light. We know that when the sun comes, most of us wake up. And then based on the availability of sunlight, the oscillator is organizing our behaviors for various activities. And for example, this Drosophila will be very active in its movement around, and we call it the locomotor activities. So basically, this concept can be taken as a central oscillator is getting some input, and based on the input, the oscillator is organizing its output. And now the question is that, is there any genetic program behind this? And there was a genetic analysis done on this fruit fly, and it revealed that, in the olden days, Konopka and Benzer in 1974 actually did a mutagenesis analysis, and they found that, if you do mutagenesis, some of the animals become arrhythmic. You can see the pattern of rhythm, that the double plot on the top, lane A, every day is, first for the normal. Here what you can see is 2 days are plotted in the one lane, and then it will be repeated again, and the first day, second day, third day, like that it is coming. So in a normal, you are seeing a normal pattern. So in that time, the animals are active, that is dark, and night time they are not active, and again they are active the next day. Like that is going, and when there is a mutation happens, this rhythm is lost. So the B is an arrhythmic mutant fly, and C is a short rhythm mutant fly, and D is a long rhythm mutant fly. And they have identified there is a gene called the period, and this now we know there are so many genes. At least there are two very core genes, per and period and timeless, and these are expressed in the clock genes in the Drosophila brain. This is a Drosophila brain expressing clock gene, and the circled area is the central clock in Drosophila brain. And I did a more systematic analysis here. You can see again, there are central clock, which expresses period, clock, cycle, bimal, etc., etc. And there are also peripheral clocks in this figure you can see. So now, and you can also see that in night, this period and timeless are expressing in very high because they are not degraded in night. And in day, by the time day comes, the light comes, these proteins are degraded by the light. Basically, timeless is degraded by a protein called cryptochrome. It is a photoreceptor photosensitive molecule present in the clock genes. When the timeless is degraded, it is a partner of period. Normally they form a dimer, and when the timeless is broken down by cryptochrome, the complex is degraded and there will be no signal. And that's how this, and when the light goes off, this protein will be again accumulating. That's how the rhythm goes on. And this can be tracked in live animals. So we have a live animal expressing this period gene and we tag it to some fluorescent proteins. And this is an experiment we have done for 7 days. So what you see in the bioluminescence is the real rhythm coming out of 4 neurons in the drosophila brain. And so that's the protein part. But on the insect, you can also see that the RNA is also rhythmic. That means the biological rhythm, the genes, for getting a rhythmic expression of genes, you have at the RNA level as well as at the protein level, rhythmic expression is integrated and it's a feedback loop. And this rhythmic expression is actually translated into the animal behavior. And on the left, I'm showing an insect of a device where you can load flies in a small glass tube, and that box can handle up to 32 flies at a time of different genotype. And then we are recording the animal activity for a month time. And here we have done 5 days of light dark experiment. Then we did the jet lag experiment. The time zone is shifting. So when the white pattern is changing, there is a 5 to 6 hour time delay for sunrise. And then after that, the animal is put into continuous darkness. And then you can see that in continuous darkness also, the animal is maintaining a rhythm. And what you have to notice in this picture is that towards the end of the day and early morning, that means, during the dawn and dusk, the animal is more active. That means they are knows that, okay, there is a sunrise coming and there is a sunset coming. So that is the beauty of the circadian clock. And with the help of photoreceptors, these animals can synchronize their circadian clock, which is endogenous and it can free run. But with the help of photoreceptors, taking input from the environment, they synchronize their activity towards the environmental conditions. And this I already explained that, the period gene is expressing in the daytime, but the light will degrade that. But during the night, as the night arrives, the protein accumulates and it forms a dimer with the timeless gene. And then they will block their own expression actually. But when the sun comes, the dimer is degraded and then the expression restarts. And I just told you a few points about how the light is integrated into clock proteins. And this is a large view of a Drosophila head. A compound eye is there through photoreceptors in that. There are 7 types of rhodopsins in that compound eye. And there's an ocelli at the middle of the head. 3 of them, they also have a different version of photoreceptor and UV photoreceptors. And inside the brain, there are other photoreceptors, something called off-power Buchner islet, and they also these clock neurons. Here you can see multiple subsets of clock neurons. The basic one is the something we call the SLNV, that is small lateral ventral neurons. All of them express cryptochrome, and this cryptochrome is integrating basically the light into the timeless and the period gene and creating the feedback loop for its cycling every day. And I also mentioned that there are multiple forms of clocks inside. We also have a central clock in suprachiasmatic nuclei, and in Drosophila, the circled one is a four neurons, and they're present on either side of the brain, which is the central clock we know from many classical experiments. But as the advances in the molecular biology came, we identified many neurons in the brain of Drosophila, clock neurons, because they express clock genes. And we wanted to know which of them are actually the master clock. So how do you test that? So we did a cloning experiment. We made a period gene usualized expressing construct, which was only expressed in the dorsal neuron. So here in this picture, you can see DNs on the top, dorsal side of the brain. And so this construct was expressing only in dorsal neurons. And we asked the, how these are network of clock neurons in the dorsal brain, how this neuron network is functioning. If you remove the central clock, the non-central clock from the peripheral dorsal neurons, can it work still alone? That's what my question. So the trick what we done is in Drosophila there are many mutants. One of the mutant is called the disconnected. That means they do not have part of the brain you can see, and that missing part do have actually the central clock. And if you cross your transgenic fly to this mutant fly, it will essentially lack the central clock, but it will have the dorsal clock. And when we did the experiment, we have found that even though this transgenic fly lack the central clock, it can still function. So the white lines is the disconnected mutant background, but it still can function its own without the central clock. And that was the discovery we have published. And this is dorsal neuron clock is also possible to connect it to the photoreceptors. And I'm just showing you some mutants background. There's something called the glass and the cryptochrome. In those mutants, you will not have rhodopsin photoreceptor and a cryptochrome mutant, but still they are able to synchronize, but the phase of their rhythm is not the same as the normal. So this is one of my publications from my PhD itself, and I was fortunate enough to be associated with the Jeffrey C. Hall. He's one among the people who got the Nobel Prize in 2017. So in summary, gene expression patterns are dynamic in life, linear or non-linear, purpose-based programs, and body plan is sketched out by normally the linear programs. You can have an exception like, your tooth is, once it is gone, it can come back again. That may be an exception. There are some cases like that. But if your body plans programs are defective, you will develop congenital developments, defects, something like cleft palate or open neural tube, etc. And the other functional maintenance, once your body is developed, the functional maintenance is mainly done by non-linear programs. You can include, your insulin secretion, your rhodopsin production, etc. It is needed every day. And some of these program errors can also cause metabolic disorders. So, and this is my slide for acknowledgements. That is my mentor Raj Sansi from University of Regensburg, and my co-mentor Charlotte, she is chair in Würzburg now, neuroscience, and Jeffrey C. Hall, no more working. And I thank for my postdoctoral work basically on the ciliopathies. Dr. Anand Surup, I was fortunate enough to work with him for an extensive time of almost 6 years. With that, I stop, and if there is a question, I'm happy to take questions. Hello.
Thank you, Dr. Sobi.
Yeah.
We can have a short discussion on this because we are running short of time.
Okay.
We are already 38 minutes late. So we can have a brief discussion, and thereafter I will call senior most professor Ram Kripal sir, Allahabad University. So before that, we can have a discussion.
Toby?
Yeah.
So is there any possibility where you can connect your circadian biology with the cilia biology? Very odd basically thing I am asking.
As of now, I do not have any direct understanding about that.
No, nobody is having. That's why I'm asking abstract question.
Yeah.
Because the cilia also function in somewhat for certain organ, not for every place, in a rhythmic pattern. Okay. So wherever the rhythmic pattern is there any connection with the internal clock or the external clock? And then you can really come up with something very new kind of thing.
See, the way to connect is, metabolic rhythms are indeed connected with the circadian clock. There are extensive literature on that.
Yeah, that's extensive literature there. So when metabolism is connected, then it is very obvious that somehow this metabolic input is coming from clock as well. Okay, or they talk to each other. But only thing people are not. Yeah.
Yeah, it may be definitely talking, but we don't have a clutch on that yet. Until we touch that and figure out where they are talking, we cannot dismiss or we cannot say that yes, it is there. We can assume that it is there. And I know that metabolism is regulated, I can say organized through clock.
Yeah.
There are so many examples for that.
So I won't take too much time because already we are running late. We will have discussion some other time. Okay. Thank you.
Okay.
Thank you, Dr. Secretariat. Thank you, Dr. Sovi, for a very nice start. You really have a very good research.
Okay, thank you very much. Thank you for your listening and invitation. Thank you.
Thank you, Dr. Sovi, for joining us. Now I request Dr. Ram Kripal sir to join us and present his talk. Before that, I would like to introduce, although he doesn't require any introduction, but as formality, Dr. Ram Kripal, he has been associated with Allahabad University Department of Physics. Dr. Ram Kripal has completed his master's in 1974 and did DPhil in 1980, after which he got appointed as lecturer in 83 and became reader in 1995 and then appointed as professor in 2003. His research experience include works on ESR and optical study of solids and liquids, on which he has been working since 1974. The area of research where he has worked include theoretical condensed matter physics. Dr. Ram Kripal received his PhD degree in physics from Allahabad University, condensed matter physics. His current research interests are in both theoretical and experimental EPR, that is electron paramagnetic resonance, also called as EPR. His research laboratory include Baryon X band 9.5 GHz, CW continuous wave, EPR spectrometer operational in the 77 to 1000 K temperature range in and Unicam 5636 UV visible spectrophotometer operating in wavelength range 195 to 1100 nanometer. There are a lot of this biography, which will take a lot of time if I go on explaining, elaborating. So, in short, regarding his biodata, I directly call upon and request Dr. Ram Kripal sir to come with his talk.
Okay, thank you, Pandeyji. First of all, I am thankful to the organizers of this international conference for inviting me to give a lecture. I will speak on EPR and optical studies of manganese 2 plus doped single crystals and nanocrystals. Slide वाला चलो. बस ठीक है. Visible है. I think slide is visible.
मैं कुछ नहीं कहता है.
Yeah, it's visible.
So I will speak on EPR and optical studies of manganese 2 plus doped single crystals and nanocrystals. The second slide gives overview of conceptual and theoretical framework. We start from quantum mechanics, atomic physics, solid state physics, then ligand field theory, and Hamiltonian theory. In ligand field theory, we study optical spectroscopy, and spin Hamiltonian theory is related with electron magnetic resonance, which includes EPR, ESR, FMR, AFMR, and so on. The right hand side.
I can barely hear you.
The cactus representation of various applications of EMR. This has applications in physics, chemistry, biochemistry, geology, archaeology, biology, life and medical sciences, bioengineering, and so on. I have used host as tetramethylammonium tetrachlorozincate. In this host, we have mixed some impurity of manganese 2 plus. So let us come to that point. Yes. EPR technique is used to obtain microscopic chemical and physical information about molecule. Optical study provides energy level ordering of paramagnetic ion, and therefore these two have been taken together. We have prepared the single crystal of manganese doped TMZ using slow evaporation method. The crystal is of orthorhombic type. EPR spectra were recorded on Varian X band EPR spectrometer. Because we are studying a crystal, we will have to record the spectrum in 3 mutually perpendicular planes, and therefore we have to decide the axes. As crystal is orthorhombic, we can take ABC as the axes for recording the EPR spectra. The spectra is recorded along these 10-degree interval. Optical absorption was recorded on Unicam 5625 spectrophotometer in the wavelength range 195 to 925 nanometer. All these recordings were done at room temperature. In case of manganese, electronic spin S is 5 by 2 and I is also 5 by 2. So we get 2s into 2i plus 1 means 30 lines in EPR spectrum. And that spectrum is you see the spectrum on the left is experimentally recorded spectrum, and on the right hand side we have simulated using a computer program called Easy Spin. Easy Spin program is available on internet, so anybody can use that with certain conditions, and those conditions are that you have to acknowledge the author. So here we clearly see that there are 30 lines. The parameters obtained are spectroscopic splitting factor G, hyperfine constants A and B, then zero field splitting D and E. E is called rhombic zero field splitting, D is called axial zero field splitting. These parameters are obtained with the help of simulation of experimental spectrum. The Hamiltonian is represented by equation 1. There are different terms, but we have neglected the terms containing F and K, as these are quite small in case of manganese. So we have considered zero field splitting, Zeeman splitting, and hyperfine structure. There are 5 groups of 6 lines, as we have seen in the spectrum. So you can take midpoint of 6 lines and plot this with respect to magnetic field. So this gives how the lines vary with field. This is given This is given in this figure. Here the center of each group varies as we vary the magnetic field. Then using these plots, we can find certain other parameters. First, we analyze the principal axis system. We take spectrum Z axis and the mid one is considered as Y axis. So in this way, we define the principal axis system of zero field splitting D. To find whether manganese has entered the host in place of some metallic ion or has gone at some interspecial site, we find different direction cosines. And with the help of these direction cosines, we have come to conclusion that manganese enters the lattice interstitially. It has not replaced Zn2 plus. So considering this interstitial site, we have also obtained the covalency parameter, and it has been found that the covalency is about 5%. After that, we come to optical absorption. Manganese is 3d5 electron system, and we have triplet and doublet. T2G is triplet, EG is doublet. These are separated by energy value, which is called dq. So if we consider different terms, then there are different levels as given as 6A1G, 4A1G, 4EG, 4T1G, 4T2G, 4A2G, 4A2GF, 4T1G doublet states. The ground is E. So transitions 6A1G, and there are 10 main bands. The spectrum is given optical spectrum 5625 spectrophotometer. A is in the range meter and B is from 190. And using this, the various bands are stable. Using this assignment, the parameters B, C, separation between doublet and triplet. This value comes out to be 670 from the value of B and C. They are 737 centimeter inverse. These are also reduced as manganese ion, and therefore we can say decrease is due to covalence. We have plotted the energy values to obtain the separation between, that is dq. This graph is about meter inverse. So combining these two studies to a conclusion that interstitially the surrounding Cl1, Cl2, Cl3, Cl, and shift from the free electron value, which is this value is, it is to say delta G is negative, and therefore there is transfer from the central line to the ligand. So this is the conclusion. Manganese enters the lattice interstitially. Crystal is covalent as B and C values are reduced, included interspecies comparison of direction cosine of G, direction cosines of different bonds. I come to the area of nanocrystal. Manganese is doped. Nanocrystal 5. So in this, we have used different like X-ray diffraction, microscopy, UV visible, photoluminescence to characterize the sample. In most of the in preparation, people use some capping agent because there is agglomeration and we do not get pure, there is clustering used, but advantage also of using capping agent. So it is better to use some materials in preparation which may be working as capping agents. So in this, ions work as capping agents. So we have used here zinc acetate and sodium sulfate. Manganese impurity, manganese acetate and remaining acetate ions work as agglomeration is avoided. After preparation, we get white precipitate is dried under vacuum at 60 degrees. The powder to have various studies. First of all, we perform X-ray and we get P position 20311 planes. This crystal has zinc blended structure. Comparison available at internet. Using this, the formula is given to 0.9 lambda upon beta. The parameters are defined. Crystallite size using this formula comes about 2 nanometer. The size is also TEM. But in TEM diagram, bit larger size. Like various crystallite in single grain. We get a bit larger size, whereas X-ray diffraction then we use UV visible absorption to obtain the band gap. Comes about 4.05 electron volts, bit larger as for which it is 3.7 electron volts. So in this way, this sample used for lead preparation. This is the optical absorption spectrum. After that, we have recorded luminescence. In luminescence, this is the spectrum. Various bands are shown here. Fixing of 4p atomic orbital manganese and there to 6A1 transition. After that, we have you will see that here gives only 6 lines which are allowed transitions, and there are 2 more lines which are forbidden transitions corresponding to delta ms is equal to plus minus 1 and delta mi is equal to 1 or 2. In case of manganese, there should be 30 lines, but because of nanocrystalline material, we get powder type, and here only we get transitions from plus half to minus half, and therefore only 6-line spectrum. This is again simulated using easy spin program. The shown in this figure, it is experimental record, and this is using simulation from easy spin program. The Hamiltonian can obtained the distribution of manganese ions in nanocrystals depend upon the particle size and surface modification. The G value is 2.0270, A is 60 in inverse, and D is 66 inverse. You can very well see that here, because there are less number of lines, so analysis is not so case of crystals. Parameter value, the structure, and here we get tetrahedral structure. In G, from free electron value, you can say that electrons are transferred. Characterization, we can find this nanocrystal. Larger band gap, so it can be used for, and it can also be used for bacteria and so on. Crystals, we can use them for near optical purposes. We can use these techniques to carry various types of samples and use them for in applications.
Thank you, sir. Thank you. We are really honored to have you in the panel and to hear you. Thank you, sir, very much for sparing valuable time for us. And we are really proud to find your presence in this panel. Now we should move ahead, and I invite Dr. D.K. Tripathi to invite the next speaker.
Thank you, Dr. R.K. Pandey.
Thank you very much.
Okay, I invite next guest speaker, Dr. Netranand ji, Associate Professor in the Department of Geography, LA School of Economics. Presently, Dr. Netranand is engaged in postdoctoral research in the field of atmospheric science and climate change in Kyoto University. He published several research papers in the journals of international repute and high impact. Since climate change is a global challenge of this era, research fields of Dr. Netranand is highly relevant to the sustainability and well-being, wellness of mankind. So I request Dr. Netranand ji, please share your research with us and enlighten the investors. Dr. Netranand ji, welcome. Please unmute.
You are
Please
muted, sir.
unmute.
Can you hear me?
Yeah.
Visual and
Both,
audio
both.
is clear?
Yes.
Okay. Good afternoon to all of you, and thank you, Professor D.K. Tripathi ji, for inviting me for this very enlightened webinar. And I have seen also your schedules and all these programs. It's very fantastic, and you have done a very excellent job to organize this type of very internationally reputed speakers also come here to speak, and it's a very nice opportunity for me, also giving me time. And now I am speaking from Kyoto, so from Japan, and today I will speak on one of very relevant topic, which I would like to just have screen share. Just I'm doing to first time. Let me do the screen share. Is it okay?
Okay, you can share. Yeah.
Dr. Netranand,
Yes.
You can share. You can share.
can you open your slides? There is an icon, share screen.
Yeah. I am clicking the share screen, but somehow okay. Desktop.
Okay, now click it again. Click on the share screen. Green button, download.
Just 1 minute, maybe. Is there any problem? I don't know. I had.
Just at the bottom, there is a green icon. Written share screen. Just
Yeah,
click
I have,
on it.
there is a share screen button and I am clicking also, but I don't know, somehow it's not going there. I don't know the problem because is it who can share, only host or all panelists? Should I
Yes,
click?
yes.
Huh?
Yes.
Should
Yeah, you can say.
You
should
can
I
share. You can share.
no, there is the option. Should I click the only host or all the panelists?
All the panelists.
All the panelists. All the panelists.
Okay.
Can share.
Yes.
Okay. I was okay. Open this.
You see screen.
Okay, Dr. Nitin, please show your slide.
Yeah. I'm just, I don't know, somehow there is maybe some problem. Just wait. Yeah, my slide I have already opened.
Yes.
Yes. We are,
Can you see?
Okay, ready. Okay.
Can you see?
Yeah.
Okay. Okay. So, sorry for this distance. Maybe there is some technical problem. So, okay. So my topic is like why the apple orchards are shifting to the higher altitudes of the Himalayas. Actually, this is a very empirical research I have done, and this paper already accepted in PLOS One journal, International Journal PLOS One. So this study, once I was in Himachal during maybe it is early 2011, and subsequently I visited 2-3 times. And when I joined here in Kyoto University around November 2018, on 28th of November 2018, I come across one news article from Times of India, Chandigarh publications, from Chandigarh. It was published from Chandigarh. That was linking this, how the apple orchards are shifting to the higher altitudes of the Himalayas. So since I have already visited 2-3 times before that, so I thought let's have done some study on this. So, and already my postdoc work is on Himalaya. So I have started working on this, and it's a very empirical and scientific one. So let's start with the topic. So as you know, maybe it is found that the apple orchards in the Himalaya have been consistently shifting towards the higher altitudes. For example, orchards have shifted to 1500 to 2500 meters in the 2000 compared to earlier elevations where it was cultivated mostly 1200 to 1500 meters during 1980s. So when 1980s, it was cultivated within 1200 to 1500 meter. By 2000, it shifted to 1500 to 2500. Then by 2014, it was again higher elevations, shifted to higher elevations like almost 3500 meters. For example, newly developed orchards of Leo village in Upper Kinnor and Kelong area of Lahul Spiti. I have already visited this village, Nako, Leo, Maling, all this area, collecting the data during 2019. And then data cataloged from different periods indicate that northward shift is due to changing of chilling hours. I mean, chilling hour is and total annual rainfall and mean surface temperature during the apple growing season. These 3-4 factors are very crucial for apple to grow and make it juice and all this. So you know what is happening on climate change, and there are many studies on climate variability, climate study. So empirically, I try to correlate all these things. Then the mean surface temperature is in all these districts has increased almost 0.5 degrees centigrade by 2014, from 2000 to 2014. I mean, within 15 years, I have observed, from the IMD temperature data, so I found that 0.5 degrees centigrade already increased. While the changing climate is reduced the apple productivity in lower altitudinal regions of the state, it is creating new opportunity for the apple cultivations in the higher altitudes, as I have observed it. So then the associated socio-economic changes are posing a new societal issues for the local farmers. Because some farmers, those who are in higher elevation now, once upon a time during 1980s or 2000, they are cultivating there only some peas, some mushrooms, and some potatoes, mostly potato and peas. But now they are cultivating apple. So they are becoming more, richer and richer because of this apple. So this is the locational map, and within this, I have taken all the 9 apple growing districts of the Himachal Pradesh. I have, during the literature survey or review of literature, I found some studies have been done, very like for Kinnaur, it was there, a study also published in PLOS ONE before, around 2012, but it is only for 1 district. So there is, for pan Himachal level apple growing case studies has not done there. So that's why I started all the 9 districts in my as a study area to see what are the changes happening in Himachal Pradesh to apple to grow, or what are the future challenges for the apple farmers. And this you can see in the map, like Una, Hamirpur, and Bilaspur. These 3 districts are not apple growing districts. There is no apple in these 3 districts. So excluding that, all other 9 like Chamba, Lahaul Spiti, Kangra, Kullu, Mandi, Sulan, Sirmoor, Shimla, and Kinnaur. All 9 districts I have taken into my research. So what are the ideal conditions for apple to grow? What are the more ideal climatic conditions? So the first, you can see the apple cultivation mean temperature is required around 21 degrees centigrade to 24 degrees centigrade during the growth period of 6 months from March to August. That means March, April, May, June, July, August, during this period. And total annual rainfall requirement is 100 to 125 centimeter. And chilling hour, which is a required chilling hour for apple to grow and juice, make it juice and very tasty, 1000 to 1500 hours of chilling hours during this 6 months period. So what is chilling hour? Chilling hours are defined as the number of hours in which temperature remains less than 7 degrees centigrade. So when temperature less than 7 degrees centigrade, then it is considered as chilling, it helps to apple to grow. So chilling hour is calculated basically on the winter season. It starts from December, January, February. That time it is helping the budding of the flowers and all this. Slowly after that it is coming. So, and this chilling hour, to calculate this chilling hour, I have applied one chilling hour model data, basically the equations. This already one published paper in Switzerland. So I have taken from the chilling hour from the National Horticulture Board of India. They have also used the Switzerland model for to calculate the chilling hours. So I used the National Horticulture of India's chilling hour calculation systems, which is basically taken from the Switzerland. So these are the equations, and here DL, CH is chilling hour. Then you can see DL is day length, day length means how many times during the day from morning to sunset, so that all this day length is calculated. Then CD stands for climatological day, then latitude, longitude, critical temperature, and critical temperature is reaching the chilling hours. And then another TS is for temperature before the sunrise and sunset for the DL. That means that is for the day length. So all these equations I have applied, and based on that, so the panel A, figure A, which shows all the elevations and all this basically is elevation DEM file. And if we'll see panel B, like here, all the 9 apple growing districts, we have taken the central point first, and then after the central points of all the districts, then we linked to calculate the maximum possibility of locations, taking each districts, to link that with particular applying that equations values on these points which you can see in the figure B, like all center points, like you can see A, B, C, D, E, all here in the 9 districts. So we have choose the center point first, and then after the center points, then maximum possible distance where we can cover. And after applying the equations, you can see here on the left side of the legends, figure legends, the years and how we have calculated the pentad. That means 5 years basis. So we started from 1975 to 79, then 80 to 85, 85 to 89, and all this type. Then we came to know, I mean, from this figure, you can see how this is shifting. We use the contour way to plot this figure. So this is just showing the spatial, how it is moving upwards. Then after doing all this chilling hour calculations, you can find within the chilling hour calculation the area, like from the 1000 to 1500 meter, you can see these are the main central districts, and here in the northern side, northeastern side, you can see where it is written like 1500 hours. So within that, it has been already come to know these are the major places where apple are growing. So then I did the temperature and rainfall analysis of all the 9 districts of Himachal Pradesh, taking data from 1975 to 2014. So you can see in almost all the districts, the temperature is increasing, and if you can see, the red line is temperature and the blue line is rainfall. You can see this is typical mountain, like bottom valley topography type, results you can see, where temperature is increasing, but rainfall is decreasing, which is not generally, generally if you say it is mountainous area, so that's why it is in hilly area, this is the features. Otherwise, generally, if temperature is increasing in some place, generally rainfall will increase, because temperature and rainfall is directly correlated, positive correlation. But here in almost all districts, you can find mostly all, there are many in mountain area, this is happening. So where temperature is increasing, but rainfall is decreasing. So these are, I have taken the IMD data, like 0.25 degree to 0.25 degree for rainfall, and 1:1 degree for temperature, grid data. So after this analysis, you can see if temperature is rising and rainfall is decreasing, so then one thing came to mind, within this temperature, within this precipitation or rainfall, whether they have considered all this, because it's IMD data, so they are considering all this, whatever possibility way to include the snowfall, amount of snowfall in the precipitation and also rainfall. When I did the primary survey, I came to know from the villagers, those who are cultivating these apples, they said me like that during early 90s or 2000, maybe 20 years before, in that particular area of Leo, Nako, and Kelang area, maybe, or even Bhormor, all this area I have visited. Sometimes snow during the winter, snowfall depth was very high, around 4 to 5 feet in 2000 or maybe 90s. But nowadays, the depth of snowfall is decreasing in this area. Maybe within 2-3 feet, it's a decrease. And there are many studies also. I have cited already that in my paper, which is already accepted, and maybe within 1 or 2 weeks, it will be online. So that I linked there. That means rainfall is decreasing. That means this is the what I have observed also from the apple growers, the farmers. What is their opinion and perceptions? Then here in this figure, specially I have plotted this. See the special variations within all the districts. You can see the panel A, temperature 1975 to the year 1975 to 1979. Then till 2014, we have made 6 pentad maps. And if you see from the lower area of like Sulan, Sirmoor, slowly if you will grow towards the higher altitudes, you can observe from the legend, you can see the temperatures. In the lower area, the red line shows, that means it's a high, temperature is high, and slowly we are going upwards. It is a very clear directions, as if it is like temperature is increasing phase-wise, like altitude-wise. If you can see the red areas, all the red signals are very low latitudinal, altitudinal areas, like within 1000 to 1200 meters. And slowly, as the blue one and the sky blue type, it is more than 6000, 7000 elevation area. So temperature trend is going in. It's generally happening during that season. And this map has been only for using the data only after growing seasons. And then in the panel B, this is about the rainfall, special distribution of total annual rainfall. Similarly, we have done all this from 1975 till 2014. All this, you can see how the situation is changing. And then from district wise, I have taken the apple production data and area and production. For example, how many, like Kullu and Manali, for example, Shimla, I mean, that area, and all the districts I have taken the area, how much area they are cultivating from the Himachal Pradesh district directorate data I have used here, and then total production, amount of, I mean, total production in metric ton. So area in with 10 hectares and metric, it is the production in 10 metric ton. That axis already in the legends you can see. So if you see the Chamba, for example, Chamba area, you can see data from 1975 to 2014. You see the production with area, it is almost, I mean, area is increasing, also production is increasing. And see in the Lahaul Spiti, it was initially from 1975 somewhere you can have some productions, but after that maybe 1981 to almost 90, it was there is almost no productions. Then there was slowly, I mean, you can see the higher growth in Lahaul Spiti is around 2000 to 2005 during that year. It was suddenly peaked and then come down. But here, Kinnor, you can say the same situations, like with area and production is increasing, because the Kinnor apple, as you know, it's a very famous one, and all this Leo, Nako, Maling, and all this area, it's very famous for upper Kinnor area. And if we come to the Kolu, it's almost area and productions is like increasing and decreasing. Then you come here with this Lahaul Spiti. I have already, here you can see how the productions area is increasing and also productions increase sometimes. So if you see the area in Lahul Spiti, this graph number 4 within this panel, you can see Lahul Spiti, how it is, and seriously, area is increasing very rapidly. And then we can see in the Mandi. Mandi is now it's within the middle latitudinal region. So most of the, very recently it's coming down, though area is there, but I think in total productions, the quality and all this is very decreasing. When we have interact with the farmers, they are very hesitant to go for apple because the quality is not good, no juicy, and it's very small in size, so it's coming down. Then in Simla and Sirmur almost now no, I mean, because of this global warming and climate change and increase of temperature, Sirmur almost it's Venice now, very low productions, and then Solan is also, you can see Solan, it's almost going towards downward. This, and then this figure is about the percentage of apple orchard area within the districts, percentage wise in different periods. You can see the legend like in 75 to 79, then 80 to 84, and all this year. You can see how the percentage in apple, percentage of apple orchards in the area. In total other productions, if we see only the apple, how much percentage it is growing. Then percentage of apple orchards in the, you can see another figure also. Then during my field survey, I have taken these pictures, photographs as a sample from the, you can see the A is from Rampur and B is from Leo village, and their altitudes is already written in the, sorry, in the, you can see in the caption, and the figure C is near somewhere in the Manali of Kullu districts. And to link that with the satellite image, you can see in the panel A of left hand side, the cultivation of apple in Leo village, Kino districts. You can see the village, and here we have taken the satellite image, and we have seen draw the picture. So here Leo village and these areas are depicting the apple cultivations. It further validate our study, like from the area and also from the figure, satellite figures. Then finally, what I came to know after taking 100 sample we have collected from the area. So this A and B figure you can see, and if you have in your mind the temperature, how the temperature is increasing from the lower altitude to higher altitudes, like it is a clear distinctions. So here you can see in the panel A, like we have 1980s altitude from 1000, all the apple growing areas like Karsog, Dharamsala, Palampur, Kutli, Arki, all this area, their altitude is written there. You can see it's within 1000 to less than 1500. That was the altitude where apple was growing during 1980s. Then by 2000, you can see the range. It is shifting towards 1500 altitudes to up to around 2200 meter altitude. So here the area is like shifted to Bharmur, Jubal, Thunang, Manali, Haban Valley, Ruru, that area it has shifted. And by 2014, you can see this is like Sangla, Kelang, and Poho, all this area it has shifted. So this is the trend of shifting because of increasing temperature and global warming. And the same things we have depicted here in the panel B, like how it is shifting towards from, you can see from altitudes having yellow color, lower altitudes within 1000 to 1500 meters, and then 1500 to 2000 or 2200 meters, and then 22 to 3100 meters elevations. And this is just similar type who is you have seen in the temperature. This is the temporal shift of apple growing. And we have applied for all this statistical analysis also to find all these links with the rainfall, temperature, Mann-Kendall test and all this, and also Z-square and all this score, which I have not mentioned here. So this is basically one study which I have done based on the empirical study, which I have visited the area and I have taken all this study, and this has been already accepted in the +1. So then I just propose how the agriculture and climate change adaptations, because along with this, I have done many other climate change impact and climate variability impact on different fields. Like my specialized area is climate change, climate variability, stochastic analysis, modeling, and all this. So I have applied like climate change adaptation, like how the climate variability influence the agriculture in Orissa, agriculture in Bihar. Like for Orissa, I have done already, that paper is already published in Climate, and another paper is published in Atmospheric Science Letters. So like 3 districts of Orissa, Kalahandi, Balangir, and Koraput, and also whole Bihar, which is under review now in sustainability. And then I have also done some climate change impact, like with my students, like how the climate change influencing the bird migration because of increasing temperature, how the birds are migrating and from Rajasthan and all this. So when we adapt that with how the climate, agriculture and climate change adaptations. So reduce the vulnerability means increase the adaptations and mitigation capacity at the same time. You need to do that. And what are the regional needs? Now identify vulnerability and opportunities with the agriculture sector. And then reduce the uncertainties, like learn from the past, monitor the present, and information for the future. So from this apple cultivations, whatever I have done, then now my next target on the apple cultivation is using the ultra high resolution model data like CMIP or CMIP5 and CMIP6. Very recently, the CMIP6 GCM data has been out. So I want to use that ultra high resolution, customize, I can customize, or we can customize that resolutions to at least maybe 5 kilometer to 5 kilometer range. And I can see the future projections, how this apple will shift further. So that type of thing, idea I'm thinking also to do in the next project. So then identify the technology to reduce the vulnerability. That means how the diversifications, then store and efficiency of use of water and genetically things. You have to think about that, because even if climate change is happening, it is all about the time scale, how you are taking the time scale. That is very important to find the empirical evidences from the different parts of the world. Then identify the institutional architecture and policy interventions to reduce or transfer the risk. So it is like giving the how institutionalize the systems for early alert and the response systems, insurance, recovery loans. Like now, if in the lower altitudinal area they will do apple, maybe they are going to get great loss. So they have to change the cropping pattern. Now they have to shift for like mushroom, potatoes, and all this area, and the plum and some other cultivations in the lower altitudinal areas. Then, so next is the like climate information systems in place. If you have the climate information to the grassroots level farmers, then it will be very good approach. They at least learn. Then bottom-up participatory process. So where the observer, they can participate and they can observe what is happening. So this is just very recent work. That's why I had thought to give one lecture on this, because it's a very recent one along with my research. So yeah, thank you all. Because I think if I have time, I can show some other works below, but if it's no time, it's okay. Hello?
Farmers are facing very challenging tasks due to unpredictable changes in climate, and they suffer a lot because of this uncertainty. You focused on the apple orchards. in Kashmir, Himachal, etc.
Yeah, I have many other studies also.
Yeah.
But
because of change in the temperature, farmers are being forced to go to the higher altitude for their cultivation and production. This is a result, direct result of the climate change. And this is not only in the orchards, apple, but throughout the continent where we are facing the same problem. Unpredictable rain, unpredictable drought condition, flood condition. So many things are happening. I think probably it is because of change in the climate. You have really talked nicely on these aspects. We can have very short discussion on this topic.
yeah, sure. If any questions, and actually I didn't check the time. If I have some time, I can discuss some other issues if there is still some time, few minutes left. Otherwise, are there question and answer sessions also?
Yeah, we can have some question. We can have for it 2 or 3 minutes.
Okay. Yeah, some of my works also, like even small 2-3 districts type. I mean, we can see how the climate change is influencing even a district level. And
Yes, Ganpati,
earlier,
do you want some?
yeah. Any question?
Hello.
Yes.
Yes.
Yes, Ganpati, do you want to raise some question?
No, nothing, sir. Nothing, sir.
Okay.
Let him continue. It is a very good topic also, and he is also very enthusiastic and wants to give some more information. So let him give that some more information, Professor Sahuji.
Okay. So actually, I have also done some climate change impact on Kalahandi, Balangir, and Koraput districts, like how the climate change influence the rainfall, temperature, and then how in that particular, because it's a mostly semi-arid regions, so how the climate change influence the cropping pattern there. So I had also done like rice and maize yield anomaly and seasonal with rainfall. So I found a very interesting study also there. Like you can, I mean, that is already published in the one journal, like climate already. So my students are engaged with that also. And so when you see the climate change, because I am attending many seminar already in the climate change, and this is my specialized area, but when I am taking personally how the climate change is happening, so what I came to know, it is a matter of time scale. How temporally you want to discuss the climate change. Because if we will tell or take 1,000 or maybe 1 millennium data of paleoclimate, then you fit these 1980s onwards, within that, you will find there is climate change as such, there is no such thing happening. It is a cyclic one. Like the same way, solar radiation coming from the earth, coming from the sun to the earth, it is fixed. It is a continuous process of 13-year cycle. Like it is going, because it takes 13 years of cyclic rotation of the sun. So within that process, it is just coming and down, coming and again, coming down and then up. So it's a cyclic type. So if we fix that within the millennium data, for example, paleoclimate, you have paper in Nature and Science and very prestigious like GRL, Journal of Climate, that type of journal, you can find many prestigious works, many prestigious journals also. So basically, how you see, since we are here now and we are observing this trend of increasing of temperature, then we are facing the problem. Maybe we never know what will happen after 100 years, 200 years, or 500 years. We never know. So when we go back 500 years before, maybe we have some phase, it is, or maybe 1,000 years before, there is some phase where temperature was very low. And now from after industrial era, temperature is increasing because of anthropogenic forcing and all this. So when you fit that within 50 or 60 years, that within range, then that is happening, the climate change is happening. And being a scientist, being a researcher, we are existing here and now we have the life systems. So we have to think, yeah, it was not there before 100 years, before 50 years, but now it is happening. So that is the way we have to understand the climate. And since we are observing these things like decreasing of snowfall, where in one area it is decreasing, another area it is increasing. So it's like, again, it's like, which area you are picking and which time scale you are fitting that curve. Yeah, that is my observations.
Thank you, Dr. Netranand. Thank you. You have delivered really very nice talk. Thank you for being with us and sparing your valuable time from Japan. Hope we will have a nice, again, we will meet sometime in next discussion and participation. Thank you very much.
Thank you. Thank you. Thank you.